Excellent. So let's go ahead and any other questions? Thank you for catching that. All right, well, today is Tuesday, October 13th. The rough stuff is behind us. Typically that endocrine and lymphatic is challenging, although again, I've graded most of the lab exams and just started the lecture exams, and so far it's looking good. So I'm very pleased actually with how you guys did on that. So that shows good preparation. I'm very happy with that. Speaking of good grades, I am incredibly pleased with your group presentations. They have all been graded. I'm assuming hopefully you've seen that in the comments that I put in there. Everybody did an amazing, excellent job. I think the lowest grade out of any of the groups was like a 44 or something like that, or 43 out of 50. So everybody did really spectacular. So I was very, very pleased with that. Excellent job from everyone. So I'm very happy that that worked. So now we get a nice, quick, pun intended, palate cleanser, in that we go from some of the more obtuse information, the hormones and our immune response, to something much more concrete, and that is the digestive system. One organ system, one rapid exploration of it, and then boom, we get our next test. So it's going to go quick, but there's a lot to cover in the next 4 days that we're going to be doing. Towards that end, there are also 3 assignments. This is one where we usually get to do some fun physiology in the classroom, digesting fats and digesting proteins and things along those lines. Luckily, we have some pretty decent lab simulators between the PhysioX and the Labster that's going to help you with that as well. So while we don't get to do the fun stuff in the classroom, you are going to get to experience those things with those activities. And then one unit review 24, which is due, as well as the pre-lab, which was due today. The test is 2 weeks from today. So again, it is going to be, like I said, a quick turnaround, but we're just doing the one organ system, so it will go relatively fast. All right, questions on any of that?
Will there be a curve on this last test?
Usually there is a curve of some sort. I don't know how extensive it'll be, but almost every test is curved a bit. Yes, so there will be. But again, I haven't finished grading them yet, so I don't know what the curve will be. But like I said, even without the curve, I think the average for the lab exams is looking like a 70%. So I'm pleased with that for this particular exam. And then with the curve, hopefully that'll be even come up. And like I said, I just got started on the lecture exams, so I haven't pulled the grades down yet or done any of those things. But I am on pace to get them done, knock on wood. I am on pace to get those done by Thursday, which would be 1 week, which is what I would hope to be able to do. All right, any other questions? All righty, then let's dive in. We are going to talk next, as I said, about the digestive system, that mystical, magical journey that cheeseburger you have for breakfast travels along its amazing path. Like all organ systems, there are 2 main groups of organs associated with the digestive system, the primary organs. And in this case, the primary organs are what we call the gastrointestinal tract, or what is also known as the alimentary canal. This is that hollow tube that goes from mouth to anus, and that we've already talked about a couple times. One of the things we know about it is the lumen of it is considered the topological exterior to the body. So remember, as we talked about, technically the lumen is outside the body, and we had a fancy word for that. I know we talked about it before. Wow, that is tiny. Let's not do that. We'll do that. I'll do that. So the lumen, as we mentioned, is the topological exterior of the body. So again, as we talked about before, if you want to get into the body, you have to pass a membrane. Me just wrapping my hand around this pen while my hand completely surrounds the pen, it is not in my body. If it wants to get in my body, it has to pass through a membrane. And it's the same thing from our digestive system as well. Just because it may be surrounded by that hollow tube doesn't mean it's in the body. So one of the unique things about the digestive system is most of the processing occurs outside of the body. When we release contents into the alimentary canal, we are expressing it out of the body or secreting it outside of the body. right? And obviously that cheeseburger looks very different coming out of your alimentary canal than it did going in. So there's a tremendous amount of modification and processing that is taking place during that. If, and again, this is one of those things we would get a chance to do in the classroom, if we had the cadaver and took the cadaver, as we mentioned, that alimentary canal is that hollow tube from mouth to anus. And in that cadaver, it would be about 30 feet long. Now, why do I have to preface that by saying in the cadaver?
It shrinks.
Does it shrink? So obviously all of us have alimentary canals. Is your alimentary canal longer or shorter than a cadaver?
Longer.
Right, so excellent. So you guys are kind of hinting at the things. So longer in the cadaver or shorter in the cadaver? Say it again. I don't remember what I asked the first
Longer.
time. Yeah, longer, absolutely. It is longer in the cadaver, right? And you guys have hit on the right idea. Death is many things, but one of the things that it definitely is very relaxing. You have your alimentary canal, and your alimentary canal has muscle tone in it. And as we know, when a muscle has tension in it, has tone in it, it shortens as a result of that. That's how it produces that tension. That's how it produces that pressure. So what happens is your alimentary canal has tension in it, and because it has tension in it, it is shorter. When we die, we lose our ATP, we lose our muscle tone, and that alimentary canal relaxes and it expands up to 30 feet. So yes, when you die, yours will expand to 30 feet as well, but yours is much shorter than that inside of your body. Now, again, we did mention that there are two main groups of organs. The primary organs, which are in this case the gastrointestinal tracts, function is what is responsible for most of the, well, let's say it this way, are responsible for all of the primary functions of an organ system. However, we have accessory organs whose job it is to help to assist in those functions. Give me some example of some accessory organs in your digestive system.
Teeth.
I heard teeth. I see gallbladder, pancreas, liver. Excellent. Give me at least one more.
Tongue.
Tongue. There you go. Perfect. Excellent. If you're not sure if an organ is part of the gastrointestinal tract or an accessory organ, then the real key is, does food pass through it? If food passes through it, like the mouth, like the esophagus, like the stomach, those are part of the alimentary canal. Right? Food does not pass through the liver, or does not pass through the gallbladder, does not pass through the pancreas, right? It may pass over or between the teeth, over the tongue, but unless you have a really large piercing in your tongue, which you then take the stud out, food's not going through your tongue, right? It may go over, it may go through your teeth, but it doesn't go through the organs. And so those make them accessories. Excellent. All right, questions on that? All right, our digestive system is involved with 6 basic functions. What are the 6 primary functions of the digestive system?
Chemical digestion.
Excellent, and I love that you said it that way, right? Because again, one of the first things that people love to say is digestion, right? Digestion, the digestive system digests, right? Yes, the building is tall because it is tall. But really, digestion in this case really means 2 things. One of those things it means, as you mentioned, is chemical digestion. What does that mean? Explain to me what we mean when we say chemical digestion.
The breakdown of the food, nutrients, and all that sort of stuff.
Okay, breaking of nutrients.
Using enzymes.
Breaking of chemical bonds.
There you go. Excellent. So I like that. We put that all together and you guys have it. It's breaking large nutrients. So let's actually add that. Using enzymes to break chemical bonds. All right. And in that way, we basically break it down into its basic building blocks. Form of simple building blocks. from macromolecules. Right? Give me an example of a macromolecule and its building blocks.
Proteins and then amino acids.
Excellent. So the big macromolecule is a protein, and we use enzymes to break those down into amino acids, right? Polysaccharides are broken down into disaccharides and even monosaccharides. Exactly. So excellent. So that is our chemical digestion. And again, one of the keys to that chemical digestion is that it uses and involves enzymes to break chemical bonds. We are changing the chemical composition of that. What's the other type? I think I saw someone write it, but it's, oh, there it is, still at the top. Alex got it. It is mechanical digestion. What does that mean?
Physically breaking down the food.
Excellent. This is the physical breaking down. However, we can really be more precise in that. Really, it's the physical breaking down of food into smaller sizes. Right? Teeth is one of the major functions that major organs that is involved in mechanical digestion, right? If I have a big boulder and I have a chisel and a hammer, and I use that chisel and hammer to break that one boulder bunt down into 75 small pebbles, right? I've changed the size of the pieces. I've broken it down mechanically, but I haven't changed the composition of it. Right? It's still little boulder pieces. Or if I have a big block of cheese and I grate that big block of cheese, right? I've changed the shape, I've changed the size, I've changed the number of pieces, but I haven't changed the chemical composition of that cheese. And so when we talk about digestion, there's really two types of digestion that are involved: chemical digestion and mechanical digestion. Both of those are going to occur in the digestive system, and they're going to occur at different locations. All right, what else? Absorption, absolutely. As we mentioned, most of the processing occurs outside our body. So once we get those small basic building blocks, we have to pass those through a membrane into our body. Bring those materials in. And do we want to do that randomly? No. We want to be specific in what we're bringing in. And from what we know, if we want to specifically bring things across a membrane, well, filtration is more pushing out, right? So in this case, this is going to be absorption that we're going to be bringing it in. But is that's going to be likely to be more of a passive process or more of an active process? Active. This is going to be primarily, mostly active process. Of course, there's that key pesky word. What's that key pesky word?
Most.
Most means not all. So there could be some passive movement. And of course, active process means that we will be using ATP. Excellent. Give me something else that our digestive system does. There you go. We don't absorb 100% of the material that gets put into our alimentary canal. So that which remains is released from the body via defecation. Excellent. Ingestion. Excellent. What does that mean? What does it mean to ingest?
Eating.
Yeah, the placing, exactly. The placing of food voluntarily into the alimentary canal. Oops. Excellent. Some argue that's the only voluntary act. That's not entirely true. Hopefully that defecation is voluntary, as is swallowing, as someone mentioned as well. I wouldn't say that ingestion and swallowing is the same thing, right? I pop a grape in my mouth, that is ingestion. I then modify that grape and then swallow it after that. So swallowing is really part of a different process that we haven't mentioned yet. And what is that? Right, motility, or really propulsion. Right? We want to be able to move the substances, right? Move the food from one organ to the next.
Peristalsis.
I'm sorry?
Peristalsis.
There you go. Oops, not sure what happened there. Absolutely. Peristalsis is one of the ways that we're doing that, but that will happen as well. I would say drinking would be an example of ingestion and propulsion as well. Excellent. There are our 6 main functions. If there was a 7th function, again, in this one, I'm not sure we can think of it as its own individual function, but really it plays a role in assisting all of the others, would be secretion, right? And again, we are going to use what type of glands here in the digestive system?
Exocrine.
There you go. Exocrine glands to release substances to facilitate these processes. Whether it is mucus to grease the wheels for propulsion, whether it is the serous fluid that contains the enzymes that are going to help to break down the foods chemically, and so on and so forth. All right, excellent. So
Is the 7th one necessary, or do we add it to the 6 basic ones?
what I would say is that it is, I guess it's necessary, but what I think, why I think we go with 6 is because secretion really, I guess you could think of as its own function, but really in the most case, it helps to support the other ones, right? Secreting enzymes and in a serous fluid allows for the chemical digestion to take place. Secreting hydrochloric acid leads to mechanical digestion. Secreting mucin, which hydrates to become mucus, helps in propulsion, and so on and so forth. So I think that secretion really helps to play a role in all these other ones. If you wanted to think of it as its own function, that would be fine. But really it helps to facilitate the other ones as well. All right. So again, here is that list that we just finished talking about. And again, notice I have them numbered 1 through 6. Obviously, some of these things have to happen before the other ones. Typically, you need to ingest food before you can break it down, or before you can absorb it, or before you can defecate it. However, is this the sequence in which these events occur? We do all the ingestion, then all the propulsion, then all the mechanical digestion, and all the chemical, and then all the absorption, and all the defecation?
No.
No, absolutely not. So many of these functions will occur in multiple locations throughout the alimentary canal. In fact, for our alimentary canal, we will take that same mystical, magical journey that the cheeseburger does, starting at the beginning when it's unset to when it then is expulsed from the body, and talk about all the processes that occur as we go through there. All right. Yeah, again, I would say it plays, secretion definitely plays a vital part in many of these functions. So again, I don't think we necessarily have to think of it as its own separate function, but you certainly can. So if you wanted to make this 7, I wouldn't have a problem with that. We are certainly going to talk about the secretions that are produced and their functions. So I don't have a problem with you talking about that as its own function as well. All right. So as we just finished mentioning, this is not a sequence of events. Some processes occur in one location or organ, but others occur throughout the digestive system, right? Ingestion occurs in one place and one place only. Where's the one place that occurs?
Mouth.
Yeah, in the mouth, absolutely. But like I said, every single organ of the alimentary canal is involved in propulsion, right? So your book's got kind of a nice figure that shows where some of the different types of processes are going to occur. And this is definitely something we are going to be emphasizing and talking about as we work our way through this mystical and magical journey. All right. Let's talk about the basic functions before we get into specifics, though. Like I said, one of the unique things about our digestive system is it is involved in processing and modifying that food, regulating the environment that technically is outside of the body. Right? And again, there's that mystical magical term where the lumen of our alimentary canal is considered the topological exterior to the body. Now, within that lumen, there's some basic processes that are going to take place, right? Our alimentary canal is stimulated by some type of either mechanical or chemical stimuli, right? When you eat that cheeseburger for breakfast, the cheeseburger expands the stomach, and that stretch of the stomach can initiate the signal. Presence of certain chemical stimuli, a change in pH, certain amino acids being present, certain basic building blocks like fatty acids or simple sugars, things along those lines are all things that can be stimuli that are going to initiate the processes. So like I said, pH, substrates, osmolarity, mechanical stretch, all of these things are going to trigger the change that is going to take place. And the changes that are going to take place in the alimentary canal are basically going to be one of two types. Either you are going to get an increase or decrease in glandular activity. So you produce more saliva or more hydrochloric acid or less hydrochloric acid. And you're going to change the motility of the smooth muscle, increasing or decreasing or even changing the rhythm of how it contracts. So in its simplest form, this is the game plan. The game plan is to receive a stimulus, and when it receives that stimulus, it modifies the behavior of the organs, where the organs either produce more secretions or they somehow change their motility. Now, of course, we need to know how this is done. And not surprisingly, there are two main ways that our digestive system is regulated. What do you think the two ways that we communicate with the nervous system and communicate with the digestive system and tell it what to do are?
Nerves.
Nervous system. And do we have any other organ systems that are specialized for communication?
Endocrine.
Endocrine, there you go. So not surprisingly, we have the endocrine and the nervous system, two different regulatory mechanisms for controlling digestion. Let's talk first about our nervous mechanism. And actually for this, I'm going to cheat and go to our whiteboard. All right. Here is our alimentary canal. All right, and right now we are, of course, talking about our neural regulation. This neural regulation really involves two reflex arcs. There are two reflex arcs that are involved in this, the short and the long. Let's talk about the short reflex first. Short reflex is what is also known as the intrinsic reflexes, because they're involved within the organ itself. Our digestive system has an elaborate network of nerves. Way back in 430, we learned a fancy word for a network of nerves. What is that fancy network of nerves called?
Anastomosis.
Plexus. Now, actually, plexus is what I was looking for. Excellent. So that one works. Involves what we call the enteric plexus. The enteric plexus is this elaborate network of nerves, which is sometimes commonly referred to as the gut brain. This gut brain, this elaborate network of nerves, is actually able to make basic decisions. Now, when it makes these basic decisions, do we want these to be big, grand decisions that are just going to affect our entire body?
No.
No. So we're going to want this to be with local effects. Basically, the way this short reflex works is there is some stimulus inside the organ. So this, again, like we talked about, could be a change in pH. It could be a stretch. It could be the presence of a certain amino acid or something along those lines. There is some type of local stimulus that occurs inside the organ. So our stomach, for instance, our stomach, we eat that cheeseburger and our stomach stretches. That stretch signal is obviously perceived by a stretch receptor, right, which measures length. So here we have local receptors. And those local receptors can be stretch receptors or chemoreceptors or osmoreceptors. Oops. Ugh. Some type of receptor that is occurring here inside of the organ. And let's just cheat and make that fit. And that signal from that receptor sends a signal. Oh, I don't like that. Sends a signal to that enteric plexus. Again, that a local network of nerves. This is not where the gut feeling comes from. Although we think of it the same way you learn something by heart, right? You learn something by heart, you get a gut feeling. These are, again, where we feel physiological changes that go on when we have these types of sensations. But obviously, all those things are occurring in the brain. Now, this is the decision where, like I said, you eat a cheeseburger for breakfast, it stretches your stomach. Your enteric plexus in your stomach perceives that information, receives and processes the information. And then, of course, like any command center would do, it then sends a signal to the effector. And in this case, that effector is going to be the local organ. And that effector is either going to be the smooth muscle of the organ or a gland of the organ. And so, of course, it's either going to, if it's the smooth muscle, it modifies the motility, or it modifies the secretion, increases or decreases secretion. And it is a local effect. Basic decisions, local effect here with the organ itself. and that is our short reflex. All right.
May I ask you a
Question?
question?
Yes.
So the neural regulation doesn't start with a hunger, right? In this
No. In fact, hunger rarely affects when people eat, right? How many people ate today truly because they were hungry? Or did you have lunch at 11:45 because you needed to wolf down that food before class started?
Okay, I
Right?
remember.
It's a little less important in a class like this because literally no one has their cameras on today. So I'm assuming it's because all of you guys are on Amazon right now and don't want to show that off. But if we were actually in the classroom, right, we would not be able to eat, right? Or people eat because they're bored, or people eat because they're stressed, or people eat because, you know, other people are eating around them and that's the time where you sit down, or when it's convenient. So this isn't hunger. This is not what we're talking about. Whether you're hungry or not, if I force a cheeseburger down your throat, when it gets to your stomach, it's going to cause your stomach to stretch. You are going to get a local effect where you're going to get an increase in motility, an increase in gastric juices to start the breakdown process of that cheeseburger.
Understand, thank you.
Yep. All right. Local effects, local stimulus. However, are there responses that can have a big effect on our digestive system? And also, does it matter? Does the food actually have to be in your alimentary canal to have an effect? If your next door neighbor, right, who used to be able to go over every Saturday night to hang out at their place because they love to barbecue, right? And he's a barbecue master, one of those that takes the tri-tip and marinates it for 4 straight days, right? Brings it up to room temperature, gets that perfect sear, and then brings it up to temperature slow and low. And as you smell that wafting over the fence and through your screen door, can it affect and modify that behavior of your digestive system? Absolutely, right? Can have a huge effect on it, both positive and negative. Right? Many people would tremendously enjoy that and their stomach would grumble as a result of that. However, what if you're a vegetarian? Is that necessarily going to make your stomach grumble and make your mouth salivate, smelling all of that burnt meat? Yeah, see, there you go. Someone else thinks it's gross. Absolutely, right? So notice in both of those cases, we're getting influences on the digestive system from something that's not even in the digestive system. Right? Just if we were to take 5 minutes and close our eyes and really think about the best meal you've ever eaten in your life, and you sat there and thought about that, would you be able to modify your behavior? Maybe start salivating, maybe you'll get some rumbling in the tummy. And I don't care how vegetarian you are, it's impossible to watch TV and see a Carl's Jr. commercial and not get hungry as a result of that, right? Exactly. Absolutely. There is nothing more delicious looking in the world than one of those Carl's Jr. cheeseburgers on TV. Not necessarily in real life, right? In real life, they don't stand up. But when you see one of those things on TV, I mean, they're just the most amazing thing you've ever seen in your life. And so the reason for this is because there is a second neural regulation. And that second neural regulation is what we call the long reflex. Right? And this one, of course, would be an extrinsic outside reflex, outside of this. And the key to our extrinsic reflex is that it involves the central nervous system. All right, let's talk about some of the differences between our long and our short reflex. And let's change the colors here to make it easier to talk about. With a long reflex, the stimulus can come from inside or outside the alimentary canal, the GI tract. All right. Obviously, it can start inside. Something like the taste, right, of that marinated mushroom, or like I said, it can also be stretch. It can also be, pH, all those other things, and etc. but here we're able to add taste to that as well, which again is a more chemoreception, but you get the idea. But it can also be outside. The GI tract. So it can be sight, smell, right? It can be just thinking about the food, right? Even top down processing. My wife, when she was in grad school down in Southern California, there was a restaurant there that had this amazing ground pepper ranch dressing that on their salads that she absolutely loved. And we would eat there all the time when we were down there. And when she got pregnant with our first child, we happened to go to there for dinner one night. And the next morning, she was really, really sick. Morning sickness had nothing to do with the food that she ate, but she threw up a lot, including that ranch dressing. And did she ever go back to that restaurant and eat that ranch dressing again? Really? After you get really sick and throw up,
No.
even if it's not related to that food? No, absolutely right. If fish tacos were ruined for Nick, right? Absolutely. I happen to know somebody who one night got really, really hammered on tequila, and they ran out of lemons to try using those as chasers, so I had to switch to oranges. And they got so incredibly sick that to this day, I don't like the taste of oranges, right? The feel of the pulp makes me nauseous. Right. So again, we have this effect where our top-down processing, our experiences with these food can actually give us an aversion to them as well. Right. So all of these things have the ability to influence it. Now, obviously, if it is coming in sight, sound, smell, or something like that, obviously this is, wow, that's huge. This is coming into our central nervous system. And how would information like that get in? So along that pathway, how would sight and smell get into the central nervous system? Cranial nerves. Excellent. Which ones? Olfactory. Excellent, which is, of course, 1. And optic, which is, of course, 2. Excellent. Right. If it was something like our memory, thinking about that, then that could be the hypothalamus. If it's a part of that taste aversion and taste, that can be our limbic system, right? Etc. All of this input coming in this way. But as Nabil also pointed out, remember too, that it can be stuff like taste. It can be stuff like the stretch receptors and pH and things along those lines. And most of that information is going to come in via what? Which nerve is going to carry most of that type of information? pH, stretch, temperature, things along those lines. Don't talk about taste. We'll talk about taste in a second. Taste is a little trickier. But most of this stuff is coming in via what type of vagus nerve? There you go, cranial nerve 10. So in that way, it is carrying this information inward to our central nervous system from those, let's see if I can, there we go. So they're being carried in from our central nervous system where it gets processed. Then once it gets processed, and once it processes that information, then that information gets sent to our enteric plexus, which then sends it to the effectors. And again, how is most of this information going to get to that enteric plexus? Can't tell if those are new answers or old answers. Pretty sure they're old answers. There we go. We'll pretend it's a new answer, or it can be. Most of it is indeed going to be cranial nerve 10, vagus nerve. Carrying that information too. Now, one of the keys to this with the central nervous system, how most of it is coming out the cranial nerve 10, is it limited to just the one organ where we experienced it? If we taste it in the mouth, does it only affect the mouth? No, because we're starting from the central nervous system and going out the cranial nerve 10, the vagus nerve. It can affect multiple organs. And in fact, it almost always will. Now, that doesn't mean the local can't affect the nearby organs, but this can potentially affect all of them. So it is a much bigger, much broader pathway when we go through that central nervous system. Again, it's still not voluntary. We're not consciously thinking, that dinner smells delicious. I should salivate as a result of it. But we have that information coming in. Right? That neighbor carrying that perfectly grilled tri-tip on a big, huge tray to share it with you across the yard from your backyard, and he trips and falls and drops it in the dog poop. Even though it smells the same, is the way your digestive system responds to that going to be differently? Yeah, absolutely. All right. So these are our neural pathways, our neural regulating pathways. Let's look at the pretty picture from your textbook. So again, we have this enteric nervous system, that elaborate network, that gut brain, with our intrinsic controls, those short reflexes where it's regulated by the enteric plexus, makes all the decisions. This involves a local cue that is inside our alimentary canal and typically gives us a local effect. We have our extrinsic controls, which are the long reflexes, where the stimulus can occur both inside or outside the gastrointestinal tract. It goes through our central nervous system. It goes through our autonomic nervous system. And then to the enteric nervous system. And notice, as we talked about, it can involve cranial nerves 1, cranial nerve 2. Anyone remember what the cranial nerve involved with taste primarily is? You don't know now, you will in a few minutes. Come on, someone remind me what taste was. Which cranial nerve provided taste sensations from the anterior 2/3 of the tongue? See, when we taught you this stuff next door in 430, you didn't think it was going to be important. You didn't think it was going to be necessary. Again, I don't randomly pick the stuff I teach you out of a hat. I teach you the stuff that you need to know. Come on, someone figure it out. Glossopharyngeal is correct in that it does provide chemosensations, more from the roof of the mouth, the esophagus, and the posterior thirds. Absolutely, it is one of the nerves involved. Glossopharyngeal 9 is one of the ones that is involved, but not the one for the taste for the anterior 2/3. Facial nerve, there you go, cranial nerve 7. Your facial nerve gives you taste, but absolutely, glossopharyngeal does as well. Spectacular, excellent. So, here, again, similar to as I was talking about, we have this representation of our short and long reflex from your textbook, where again, the short reflex is all local, all contained within the alimentary canal, whereas the long reflex involves the central nervous system and the autonomic nerves, can start inside or outside the alimentary canal. And one of the things that always bothers me about this particular illustration is, again, notice, I understand why they put it here, but they have put taste here. Where does taste take place? In the mouth, right? So again, that is part of the alimentary canal. So really, that part should be down here as part of a stimulus of the alimentary canal, because the mouth is part of the alimentary canal. But sight, smell, thought of the food, right? Previous experiences, all those kind of things as well. All righty. Now, that is the neural control. let's talk about hormonal control, although we're really not going to talk about hormonal control because there are over a dozen different hormones that are involved in digestion. And I'm not just going to sit here and rattle them all off. But we will talk about the general functions of our hormonal mechanism. The mucous membrane of our alimentary canal contains many single-celled endocrine glands. Right? We've seen single-cell exocrine glands before, like the goblet cells that we talked about. Well, here our mucosa contains many single-celled endocrine glands called enteroendocrine cells. And again, as I mentioned, they produce well over a dozen, well over 18 different hormones. I'm not going to list all 18 for you. We're not even going to talk about all 18, although we will talk about some of the important ones. Obviously, these are circulating hormones. So as we talk about, the advantage of that is they're longer lasting. They can get in the blood and they have the potential to go anywhere they need to in the body. So not surprisingly, they allow us to coordinate the activity of many organs in the alimentary canal, as well as accessory glands and organs as well. And being hormones, they can influence other systems as well. In fact, our friend histamine is an example of a hormone that is used in our digestive system, and we've also talked about it in our immune response as well, and there are others. All right, but again, the effects these are going to have, that's what I want to say, are primarily the same two effects. We are going to modify the mechanical movement of the organs and modify secretion. So those are the things that are going to be influenced, and that's the same. And what we'll do, as I said, as we work through our glands, we'll talk about individual hormones and what those individual hormones do in the different locations where we talk about them. All righty. Questions on that? All right, excellent. Now that we've talked about the basic function, what we need to do is talk a bit about the basic anatomy of the alimentary canal. It is a bit early for this, but this is a good natural stopping point. And like I said, normally we have a lot of physiology in the class, so I don't have quite as much lecture to go through. So I think this is a good time to go ahead, even though it's a little early, and take our first break. We will take our first break, a 15-minute break. Looks like it's 12:50, so we will restart at 1:05, and we will pick up the lecture from there. Right, because we got a lot more anatomy and vocabulary to talk about coming up. All right, so any questions before we take our first break? All right, excellent. I will see you guys in 15 minutes. Let's go ahead and get started. So our goal is now to talk about, as you can see here, the basic anatomy of the alimentary canal. So again, I think a good starting point for this would again be to go to our whiteboard. Go ahead and save that one so I can clear it. There is the basic anatomy of the alimentary canal basically goes from the distal third of the esophagus to the rectum. Oops. There you go. Excellent. Notice this does not include the oral cavity, the pharynx, and the proximal 2/3 of the esophagus. And it also does not include the anus as well, right? If you think about those two locations, right, this does not include the openings. Let's just say it that way. The entrance and exit of the alimentary canal is specialized. It's in anatomy, but the majority of it, so, 27, 28 of the 30 feet has this same basic anatomy where there are 4 main layers to the alimentary canal. And I think it's easiest to go from the lumen of the alimentary canal to the outside, or again, what is primarily going to be the abdominal pelvic cavity. I know that's not entirely true if we're talking about things like the esophagus and stuff like that, but for the most part, this is the basic anatomy. So let's start at the lumen. The layer that faces the lumen that is open to the outside world, of course, is what? Lines, surfaces that are open to the outside world.
Mucus.
Yeah, exactly, our mucous membrane. Or remember, mucosa. Mucosa is the noun, mucous membrane. Mucus is the adjective that describes the noun, membrane. Fun, excellent. And as of course we know, all mucous membranes are comprised of an epithelial tissue that sits on top of a connective tissue. And anyone remember what epithelial tissue it would be that lines the alimentary canal from the distal third of the esophagus through the stomach, through the small intestine, through most of the large intestine?
Simple columnar.
Simple columnar, excellent. It is a simple columnar, often, not always, but often with microvilli. Right, not all of it's going to have microvilli, but many of it is going to, much of it is going to. Oops. And what connective tissue would it be sitting underneath that?
Lamina propria.
Well, careful, the tissue type is an areolar connective tissue, right? That is the connective tissue for, but you are correct. This particular, an areolar connective tissue that is part of a mucous membrane, remember, based on location, we give a special name to it. And that special name, indeed, as you mentioned, is the lamina propria. Again, it's one of the things that gets us in trouble sometimes on the exams, right? We have the information, it's even correct information, but it doesn't answer the question. So you got to be careful. If I ask for the tissue type, then I definitely want areolar connective tissue. If I want the tissue based on its location, I want lamina propria. All right. There you go. Excellent. We know that is absolutely going to be the two layers of the mucosa, except in this case, in our alimentary canal, it turns out there is a third layer to the mucosa. And that third layer to the mucosa is a thin layer of smooth muscle. This thin layer of smooth muscle is known as the mucosa muscularis, or muscularis mucosa. Oops. So what we actually have here is this mucous membrane. So I'm going to cheat and move that down there for now. That will allow me to grab this and do some of that drawing that I love so much. So we will start first with a simple columnar epithelial tissue. Pretend these are all uniform, although they're not all going to be perfectly uniform, but you get the idea. There we go. We have our simple columnar epithelial tissue, and that's taken too long, so I'm going to cheat and make it shorter. There we go. And it sits on top of an areolar connective tissue that we call a lamina propria based on its location. And that sits on top of a layer of smooth muscle. That is the third layer of our mucosa. Now, let's talk function. What is the function of the simple columnar epithelial tissue? Especially if it has microvilli. Oops, no.
Secretes mucus.
True, absolutely. One of the things that it is going to do is it can secrete mucus. As we know, there may be individual cells located within this, those goblet cells that can release mucin. What else is it going to do?
Absorption.
Absorption, absolutely. This is where the absorption is going to take place. And of course, here inside of our areolar, we will have the small capillaries for that absorbed material, also maybe some small glands to help in that secretion. So we have that as well. But what's the purpose of this thin layer of smooth muscle? What would it do? Why would we want a thin layer of smooth muscle?
Peristalsis.
Peristalsis is going to move that entire cheeseburger through our alimentary canal. Do you think a thin layer of smooth muscle, just that's part of the mucosa, is going to help us to be able to do that?
Maybe not.
Probably not. Now remember, it's not connected to the microvilli, but I like your thinking. Absolutely. While this may not be able to allow for major movement inside of the alimentary canal, what it is going to be able to do is change the shape of the mucosa. So for instance, helping to increase or decrease surface area. Or, right, instead of a cheeseburger, maybe you have that peanut butter sandwich for breakfast, right? And you get a little bit of that peanut butter stuck on the roof of your mouth. What do you need to do? Well, you need to use your tongue to pry it off of there. Well, maybe it gets stuck to the alimentary canal instead. So a little bit of contraction can help to dislodge anything that gets stuck to it as well. So we can dislodge materials or change the shape of the mucosa to increase or decrease its surface area for to facilitate absorption. Excellent. Perfect, perfect, perfect. Now, underneath this, whoops, nope, didn't want to do that. The second layer, so again, let's go back just so that we don't get lost because I'm doing more writing than I meant to. We have our mucosa there, perfect. So now we can do this. Deep to the mucosa, we have a layer called the submucosa. The submucosa is primarily composed of areolar connective tissue. So even though I'm drawing it here in blue, is there really any difference between the areolar connective tissue that is a part of the submucosa and the areolar connective tissue that is part of the mucosa? Are they the same or are they different?
Same.
Same. They're both the areolar connective tissues. Does that mean this one is a lamina propria?
Yes.
No.
Oh, no.
Yeah, a lamina propria is only an areolar connective tissue that's part of the mucous membrane. This is below the mucous membrane. This is not a part of the mucous membrane. So basically on the other side of this smooth muscle wall is more areolar connective tissue, but it's not a lamina propria. This region, though, because it's an areolar connective tissue, has a lot of space in it. So there's going to be space for things like large blood vessels, right? After all, if I have those capillaries collecting the absorbed material from the lamina propria, I need to send them to the liver. So I need large blood vessels. These will be where large glands will be located in this region. And a portion of our enteric plexus is going to be located in here as well. So part of that elaborate branch of our nervous system, that branch of the enteric plexus is going to be located in here. And this is the submucosal plexus. Submucosal plexus is one of the branches of the enteric plexus, one of the sublayers of the enteric plexus. And guess what it controls? Well, if it's in the submucosal layer where the large glands might be located, and the two functions of our enteric plexus are to increase motility and modify glandular behavior, What do you think the submucosal plexus does?
Stimulates glands.
Yeah, it controls the glands, absolutely. Its job is to control the glands. Excellent. Perfect. All righty. Now, deep to the mucosa is a layer known as the muscularis externa. Now, you will sometimes just hear this being referred to as the muscularis. If you just use the term muscularis, I will not mark you wrong for it. I do think saying muscularis externa helps to differentiate it from the muscularis mucosa. But like I said, you will see in the literature that sometimes it is just referred to as the muscularis. So I won't ding you for that. But this muscularis mucosa is comprised of two. There we go. This is what I want. Two layers of smooth muscle. The superficial muscle, and again, by superficial, in this case, I mean closest to the lumen, is going to be a circular layer. Of smooth muscle. And the one that is deepest, furthest from the lumen, is a longitudinal. Oops. So basically the first layer, and I'll cheat and draw them this way because we would see them. So I'm going to draw them this way for right now, but I'm going to cheat and extend one of these out, how basically it would come up and around and down through the back of the organ that way, would be the circular layer. And in this particular view, in this particular sectioning of it, would that be a cross section or a longitudinal section of that tissue? What is the shape of smooth muscle cells?
Elongated.
Cross.
Yeah, they're elongated, they would be elongated spindle-shaped structures. So what you're looking at right now is a cross section through that layer that way. And that is what the circular layer would look like in this case. And then perpendicular to it, you are going to see a longitudinal layer. And so that longitudinal layer basically runs the length of the organ. So we have a circular layer and a longitudinal layer. What is the function of the circular layer? What does a circular layer of muscle around a hollow organ do?
Constrict.
Well, okay, I like the idea of, well, obviously all contracts. Yeah, there you go. It's going to change the diameter. It's either going to constrict or dilate. It's going to either contract or relax. And when it does that, it is going to change the diameter of the lumen. So the function of a circular layer is going to change the diameter of the lumen. And what would the function of a longitudinal layer be?
Peristalsis in this.
Well,
Changes
again,
the length.
say again. There you
Changes
go.
the length.
Changes the length of the organ. Excellent. So you've got a piece of alimentary canal, and when the longitudinal layer contracts, it gets shorter. When it relaxes, it gets wider. So this is changing the length. Absolutely. So we have these two distinct layers. One of the key points, and I'll cheat and sneak it in up here, when we talk about this circular layer, in some areas, this layer enlarges. So much so that it becomes so thick that when it contracts, instead of just changing the shape of the diameter, it can actually close the diameter, basically forming a valve-like structure that can regulate the flow of movement. And when this circular layer enlarges, what's the fancy name that we give to that valve?
Sphincter.
There you go, sphincter, perfect. Helping to regulate the flow of our food through our alimentary canal. Excellent. Now, you are correct. These muscles individually change the diameter, change the length, but working together, they are going to help to, for instance, propel food through by peristalsis. In fact, peristalsis is a rhythmic contraction of these two layers. Basically, what happens is we squeeze the diameter, then we shorten the length, and squeeze the diameter and shorten the length, and squeeze the diameter and shorten the length. And as we do that, we are able to propel that food through our alimentary canal. However, if we change the motility, change the sequence of the contractions, we can do other types of movements as well. And we'll talk about that in just a minute. All right, before we do though, there is one more layer. What do I need? Green, blue. So let's go purple. The deepest layer from the lumen is a serosa. Or what we call a serous membrane. What do we know about serous membranes?
Right.
Apparently nothing. What do we know about serous membranes?
They prevent friction.
Excellent. I like what I've seen. They make transudate, they are thin, they reduce friction. They line cavities that are closed to the outside world. All right, produce that serous fluid. Excellent. Luckily, we know exactly the anatomy of a serous membrane. So once again, it is going to be an epithelial tissue sitting on top of a connective tissue. In this case, the connective tissue is going to be deep and the epithelial tissue is going to be superficial to the space of the abdominal pelvic cavity. And what would the connective tissue be of a serous membrane? Excellent, areolar. Connective tissue. Is this a lamina propria? No, because it's not part of a mucous membrane. Well, someone mentioned it needs to be thin. So if we want to make this as thin as possible, what epithelial tissue are we going to use?
Simple squeeze.
Simple squamous, excellent. Epithelial tissue. Now, this simple squamous epithelial tissue does get a name based on its location, and I'm going to abbreviate it this way, BOL, for based on location, so period there. Based on location. Based on location, what do we call a simple squamous epithelial tissue that is part of a serous membrane? Well, we'll get to transudate in a minute. You are right. We'll get to transudate in a moment. But again, very similar to what we saw in the heart. There we go. Mesothelium is the tissue. And that transudate, remember, is the name based on location that we give for the thin layer of serous fluid that lines the membrane, right? Based on location, we call that a transudate. Excellent. So, as you guys have pointed out, we get a little bit of areolar connective tissue. A little bit of areolar connective tissue. We get a simple squamous epithelial tissue, one layer of flat cells. That is a mesothelium based on its location. And then on top of that, we get a thin layer of serous fluid that is that transudate. At least that's what we get most of the time. Many of the organs of the alimentary canal, or portions of the organs of the alimentary canal, have this anatomy. However, not all of them have a serosa. Some, and for this I'll use orange, some are going to have what we call an adventatia. That's all right. There we go. Adventatia. So either going to have a serous membrane or an adventatia. If you have an adventatia, then it is just a fibrous connective tissue. And what it does is it anchors the organ in place. So notice with a serous membrane, that organ is going to be able to move around. We want to reduce the friction. But some of the organs we want anchored to the wall. We don't want them moving around in the same way. And so in that case, they will have an adventatia. And we'll actually figure out which organs do which in just a minute. All right. Fun with vocabulary. Questions on this?
Yeah, you drew the longitudinal layers of the muscularis externa, like you drew 3 of them. Is there 3 longitudinal layers?
No, what I was trying to emphasize is those were individual cells. Those are the plane of section for the individual cells. Let's cheat for a second and go back to the picture from your textbook. Notice here, when we look at our, and I'm going to switch to the highlighter here so I don't mess it up too much. Here are the 2 layers. This is layer 1 and this is layer 2. Notice in this particular plane of section, the circular layer has been cut in cross section and the longitudinal layer is in longitudinal section, right? Because the 2 layers are always perpendicular to each other. However, notice if we had cut the slide differently, then it would be possible to see the circular layer longitudinally and the longitudinal layer in a cross section. So what I was just trying to show is that in our 2 layers, the cells are going to be perpendicular to each other. So I was just trying to show multiple cells in cross section and multiple cells in longitudinal section. But notice if we cut the stomach the different way instead, then what's cross section and what's longitudinal section can change. So again, one is a plane of section and one is the layer and the layer type. Okay?
Gotcha. Okay, thank you.
Yeah, no, it's a great question. I'm glad you asked that. All righty. Any other questions? Because like I said, it's about to get worse. Or hopefully better. The goal isn't to make it worse. The goal is to make it better. All right, perfect. So we've got this picture. I'm going to go ahead and save it because I want to do one more picture as well. And then we'll go back to all the pretty pictures in the textbook and do this all again. Because one of the things that I want to emphasize is, again, when we take an organ like the stomach, and let's draw a really simple stomach here. Esophagus comes down, it goes around that way, comes out like that. Basically, if you think about it, the same way we were able to cut through the heart and see all the different layers, that's what we see here as well. And I'll just do it in words this time because it'll be easier, right? Notice here, if we think about it, facing the inner lumen, so here is our lumen. Here we are going to have, obviously, a thin layer of mucus. Oops, so I spell it right. Under that is going to be our simple squamous our simple columnar epithelial tissue. That is sitting on top of an areolar connective tissue. which is sitting on top of the muscularis mucosa. All right. Underneath that, we have more areolar connective tissue. Underneath that, we have 2 layers of smooth muscle. And then on top of that, we have more areolar connective tissue sitting on top of a simple squamous epithelial tissue. Which on top of that has a thin layer of serous fluid. All right. And then the outer wall. We have our space here, which is the lumen of the stomach. We have the space out here, which is our abdominal pelvic cavity. All right. And we have names for all of these things. This together, all right, we call what? Mucosa. Excellent. All right, our mucous membrane. We have that mucosa. Excellent. This areolar tissue by itself, what is this? Submucosa. Ah, nuts. All right, excellent. These 2 layers of smooth muscle, what is the superficial one? The closest to the lumen?
Circular.
Circular. And the one furthest from the lumen, closest to the abdominal pelvic cavity?
Longitudinal.
Excellent. Remember also in our areolar connective tissue, we had that submucosal plexus that we said controls the glands in the submucosa. Well, here we have smooth muscle. Doesn't smooth muscle need to be controlled? Yeah. So as I forgot to mention is here in between the 2 layers of the circular and the longitudinal layers of the muscularis externa, we have the myenteric plexus. And this myenteric plexus is what controls the smooth muscle activity. So these 2 things together, the submucosal and the myenteric plexus together form our enteric plexus, what we called our gut brain. And I already mentioned it, so I'll go ahead and put it here. This is our muscularis externa. Needs to be red. Perfect. And then all of this right here, the areolar, the simple squamous, the serous fluid, what is that all collectively?
Serosa.
Serosa, excellent. Is there something else we could call it?
Visceral.
Okay, there you go. Based on its location, we could call it a visceral what?
Peritoneum. Oh my goodness.
Peritoneum. Hey, as long as you spell it, I don't care how you say it.
Yeah.
Excellent. This is the visceral peritoneum. Excellent. All right. And then just for the heck of it, I think we have a couple more locational names. So things that we name based on their location. Let's cheat. I'll move this back here a little bit, move this back here a little bit, move this back here a little bit, which will give me a chance to sneak in here some names based on location. And so starting from the top, what things have names based on their location? Anything in the mucosa have a name based on its location?
Areolar connective tissue.
Yeah, the area connective tissue is the lamina propria. Excellent. Oops. Excellent. Not that, not that. Ah, simple squamous epithelial tissue. What did we call that?
Mesothelium.
Ileum. Excellent. And the serous fluid on top of it, what did we call that?
Transudate.
Excellent. And like I said, you could argue, again, the serosa, when it's there, is visceral peritoneum, which is another name based on its location. Excellent. So stomach, mucous membrane on the inside, serous membrane on the outside, smooth muscle in between. Basically, that's its anatomy.
I have a question. Did you say the myenteric plexus is between the submucosa and the smooth muscle?
No, the myenteric plexus, great question. The myenteric plexus is between the circular and the longitudinal layers of the smooth muscle.
Okay.
It's not mentioned on the picture in the book, right?
It is on the picture in the book. Notice, highlighter, yellow fibers here within the submucosa is our right, submucosal plexus. And then here, these yellow axons between the circular and longitudinal layers is the myenteric plexus. So which together form that intrinsic enteric plexus. So it does actually show it nicely in the picture.
Oh, yes. Thank you.
Yep. And in fact, the picture shows one other pretty thing that we need to be able to identify. Right? And this comes back to that very last based on location name we did, a visceral peritoneum. This serous membrane, and we'll go ahead and use, what color haven't I used yet? We'll use the purple here for this. Right? As we know, this visceral peritoneum lines the outer surface of the organ, but we also have a serous membrane that lines the cavity. And what do we call this serous membrane that lines the cavity?
Right.
Parietal.
There you go, excellent. Oops. Peritoneum, excellent. And it happens to have the exact same anatomy, simple squamous epithelial tissue, serous membrane. And if you remember back in 430, notice we've got kind of a coronal view this way. But if we were to cheat, and down here, I'll sneak in a cross section. Actually, I don't want to use green. Let me use black. So here is a cross section through the abdominal pelvic cavity. All right. Here is a cross section through the stomach. And as we just finished talking about, we know that the visceral peritoneum wraps around the organ and that the parietal peritoneum wraps around the cavity. But we also learned that these two membranes need to connect to each other. So if you remember back in 430, we talked about how this membrane, the parietal membrane, comes off to go to the organ, off of the abdominal pelvic cavity, come up to the organ, wrap around it, and comes back. And when it does that, it forms a double layer of serous membrane. This double layer of serous membrane houses the blood vessels and the nerves that go to the organ. And does anybody remember the name we gave to this double layer of serous membrane, or double layer to be more specific, double layer of peritoneum? that connects, oops, so I spell things right. Anybody remember what we called those structures, the general term for that structure? Excellent, Vanessa's got it. A couple people got it, Ludmilla as well. Mesentery. Excellent, so it's the mesentery. Or plural, mesenteries. Excellent. And now we will actually identify the specific mesenteries that are going to be found in the organ as well. And in fact, if we go back to our pretty picture from the textbook, lo and behold, you see how here the serous membrane, made up of a simple squamous epithelial tissue and a relar connective tissue, wraps around the organ and then comes off. Wraps around the organ and comes off, basically forming that double layer of serous membrane, housing blood vessels, nerves, lymphatic vessels, all of those things. And this structure here is that mesentery. So notice everything that we've talked about, everything that we saw in the drawing, this picture from your textbook does just as good of a job of showing it as well. All right. So let's go through the pictures from the textbook, make sure that this all makes sense. If you don't like my drawings, then here we have some pretty drawings that will show this as well. Again, starting from the lumen and work our way out, we have our mucosa that is made of really three, and let's be more specific here. Sublayers. So we're more precise in our wording here. So our mucosa has three sublayers. What are those three sublayers? Most superficial one is the
Epithelium.
excellent, the epithelium. Absolutely. What type of tissue? Sorry, I closed the chat window. There it is. Yep, excellent. Simple columnar epithelial tissue. Perfect. Underneath that, we have the areolar connective tissue containing the lamina propria. Notice that lamina propria can contain small glands. What do you think that green thing is in the lamina propria?
Mucosa-associated lymphoid tissue.
Excellent. Or more specifically, that would be a lymphatic follicle, right? So we'd have that lymphatic follicle in there, a little bit of lymphatic follicle. And if this happened to be the ileum of the small intestine, what would the fancy name we would give this lymphatic follicle be?
Peyer's patch.
Peyer's patch, exactly. Especially there were a lot of them clustered together. Absolutely. Excellent. And then deep to that, we have that muscularis mucosa that is able to change the surface area, make minor contractions to dislodge any food that may be attached to that mucous membrane. Excellent. And we did all the functions of that. Submucosa, again, more areolar connective tissue, but notice with larger glands, larger blood vessels, and that submucosal nerve plexus, which controls those glands. Next, we have our muscularis externa, or again, remember we said we could just call it the muscularis. I'm fine with it either way. But muscularis externa does help to distinguish the two. Made up of two layers. The first one is a circular layer that is going to change the diameter of the lumen. And the second is the longitudinal layer that is going to change the length of the organ. And we talked about it, but here is the picture where again we see that myenteric nerve plexus. So these two branches collectively form our enteric, or that intrinsic nerve plexus. Myenteric controls the smooth muscle, submucosal controls the glands. Now, as we mentioned, we are going to use these smooth muscle contractions for all sorts of things, but one of the primary things it gets used for is peristalsis. as we talked about, we get rhythmic contractions. And this is a simple picture, but I think I have a prettier one. Here we go. I like this one. So notice what we see here is first a contraction of the circular layer, and then we shorten the organ, and then contract the circular layer and shorten the organ, and contract and shorten, and contract and shorten, and contract and shorten. And as we continue to do that, constricting and shortening the organ, we move that food from one location to another in the alimentary canal. So peristalsis is not the only, but it is the primary way of propulsion in these hollow organs. In fact, almost all of our hollow organs, really the blood vessels that we started with are really the only exception. We saw lymphatic system, though they're large vessels, use peristalsis. Urinary organs are going to use peristalsis. We're going to see peristalsis in a lot of different locations. This is one of them. However, peristalsis isn't the only type of muscle movements that can occur. Notice this simple illustration. It gives an example of a type of muscle movement that takes place in the small intestine called segmentation. What happens is one part of the alimentary canal contracts and then the next, and they alternate. And notice by having just the circular layers alternating back and forth, notice what happens to the food. Is the goal here propulsion? Is it getting the food from one end to the other? Is that the primary goal of this? No. Some minor movement can be occurring, but in this case, it's squishing the food back and forth between those two areas. Why might that be useful?
Giving more time for absorption.
Not only just to give it more time for absorption, but also if you think about it, it's rubbing it back and forth on the small intestine as well. And if you rub something back and forth on it, that basically increases the surface area. So that absolutely is going to increase absorption, make absorption easier. Why else might this be useful? Yeah, it plays a role in mechanical digestion. I understand why you guys had a hard time with that one. I know every single person in this class takes a bite of food and chews that bite of food the 25 times you're supposed to chew it before you swallow it down. I know everybody in this class does that. But believe it or not, there are people who will take a bite of a sandwich, chew it twice, and swallow it down. I know it's an appalling thing to think of, but it does actually occur, right? So for those individuals, luckily, our alimentary canal is able to increase the mechanical digestion by being able to do this segmentation. Mixing it back and forth also mixes it with the enzymes. That's going to help to facilitate chemical digestion as well. So notice these are important mechanical movements, but they're not just involving and moving it from point A to point B as well. And there are others. We're not going to talk about all of them right now, but in our large intestine, we get mass movements, we get haustral contractions, we have those valves that we'll talk about. So there's a lot of smooth muscle activity that is going on in our alimentary canal. Lastly, we have our outer layer, which is the serosa. Again, serosa is synonymous with visceral peritoneum, an areolar connective tissue, a simple squamous epithelial tissue, and a thin line of serous fluid called a transudate. And again, it connects to the parietal layer via our mesentery. And we'll identify those mesenteries in a minute, like I said, except when it doesn't. In a few instances, these organs don't hang inside the abdominal pelvic cavity. Instead, they are anchored to the wall of the abdominal pelvic cavity. And when they're anchored to the wall of the abdominal pelvic cavity, they're done so by an outer layer known as an adventitia. Let's take a look at the mesenteries, then we'll see which organs aren't and what we call that condition when they are anchored to the posterior wall of the abdominal pelvic cavity. Well, you might actually know it. When an organ is connected to the posterior wall of the abdominal pelvic cavity, it's not in the abdominal pelvic cavity, it's really behind the abdominal pelvic cavity at that point, or in the back wall, or really behind the peritoneal cavity. And so what would we call something like that? Anyone know? There you go, retroperitoneal. Excellent, perfect. So let's talk about these. All right, our mesenteries, as I mentioned, are a double layer of peritoneal membrane. Has a little bit of areolar connective tissue in between, and it is a passageway for blood vessels, nerves, and lymphatic vessels. Mesenteries is the general term, but as we know, our anatomists love to name everything. So they're going to give specific names to specific mesenteries, and you are absolutely positively responsible for this. This helps to stabilize the organs in the abdominal pelvic cavity, but still allow for movement without entanglement. Right? Back in ancient times, there were things called earbuds. And earbuds weren't these wireless things that you put in your mouth. You had a little, don't put in your mouth, don't put them in your mouth. That you put in your little box, and they, connected to anything in the house. And if you lost one, it would beep and you could find it. Back in ancient times, the earbuds had cords. And you could actually take your earbuds with its cords, put it in your pocket, and immediately take it out. And that cord would be incredibly tangled up, and it would take you an hour and a half to untangle. We obviously don't want that happening to our small intestine. So by stabilizing it with these mesenteries, we are able to allow it to move, but provide some structure and stop it from twisting itself up. But like I said, it doesn't cover all of the abdominal pelvic organs. Some of them are actually behind the peritoneal cavity. And as mentioned, we call those retroperitoneal. So let's look here at some of the individual mesenteries that you are absolutely positively going to be responsible for. Notice organs like the liver, like the stomach, like this portions of the small intestine that are found within the abdominal pelvic cavity. We call these intraperitoneal organs. And it is the intraperitoneal organs that are surrounded by a serosa, that visceral peritoneum. So the stomach is a great example of this. Notice we see the stomach here, and in light blue, so we'll just emphasize the light blue, that is that visceral peritoneum wrapped around it. Notice our transverse colon has some of that visceral peritoneum. Our small intestine has the visceral peritoneum. All this light blue represents that visceral peritoneum. And notice the liver has it as well. But notice also there is 2 layers of mesentery that actually connect the liver to the stomach and actually support the stomach superiorly. This is known as the lesser omentum. Notice off of the inferior portion of the stomach, there is a long drape-like mesentery that drapes down, almost down the entire abdominal pelvic cavity, and then comes all the way back up and connects to the transverse colon. This is called the greater omentum. The transverse colon itself is anchored to the back wall of the abdominal pelvic cavity by the transverse mesocolon. And the small intestines are held in place by a big elaborate sheet of mesentery that is called the mesentery proper. Your book sometimes refers to it just as the mesentery, but for clarification, to be specific, when we are being specific about the mesenteries for the small intestine, we will call it the mesentery proper. Mesentery is a generic term. We will only use it as a generic term. Mesentery proper is the specific name for the mesentery associated with the small intestine. We see this really nicely with the sagittal section here. Notice here, they've done this great job of holding up the greater omentum. So here we see the greater omentum being held up. That does two things for us. Remember, it goes off of the stomach, comes around to the transverse colon. So we see where it connects the transverse colon, but we also see where the transverse colon is connected to the back wall, and that is called the transverse mesocolon. And here we see that mesentery proper that is holding our small intestine in place. And this also allows us to see one more mesentery we haven't identified yet. We'll color it green. Notice here is this S-shaped portion of the colon known as the sigmoid colon. And this little bit of mesentery that anchors it to the back wall is the sigmoid mesocolon. So these are all of the mesenteries you are responsible for. I think I have one more picture. Excellent. This one does a nice job of showing that lesser omentum that connects the stomach to the liver. I will tell you one more cheat for this. Great question. I'll answer that in a second. Let me finish this, what I was saying here, and then I'll answer that because that is an excellent question. One cheat to help you to remember this is notice our stomach has this concave and a convex surface to them. This concave curvature is known as the lesser curvature. And notice it is the lesser omentum that connects to the lesser curvature. Here is the greater curvature, and that is where the greater omentum would come off. And again, notice it's been cut, but it would drape down over all the top of this. Excellent. So now let's talk about what happens, and let's actually cheat and go back a picture. Two pictures. With a hernia, let's take a simple one, an inguinal hernia. An inguinal hernia is basically where there is a weakening of the muscular wall. And when there is a weakening of the muscular wall, yeah, any organ, any animal that you dissect would have some mesenteries inside of it. The rat is a great example, fetal pig. I can say I've never actually dissected a chicken, but my guess is that it would have mesenteries as well. Again, inside the abdominal pelvic cavity. But with that hernia, that weakening of the wall, the increased pressure basically can force things out. Sometimes what comes out is just a little bit of the fluid. The transudate can express out as a little bubble into this space. But it is possible that a piece of the mesentery can actually extend out into the space, typically the greater omentum. And in some instances, it could actually be a piece of an organ, like a piece of the small intestine could be expressed out that weakness and that weakened hole as well. So it isn't so much that the organs separate from the mesenteries, it's that the mesenteries allow these things to move so that when there's an increase in pressure and a weakness in the wall, those things can be expressed out that opening. Most commonly, it's fluid or a bit of the mesentery, especially that greater omentum, which hangs in front. It's much more dangerous if it's like a piece of the small intestine or something that projects out into that space, because it can actually get restricted and stop the flow, and that can actually cause tremendous pain and discomfort and dangers associated with that. All right, those. Excellent. However, I also, if you remember, we've got these great resources that you should be taking advantage of. Let me do this first. Where'd it go? There it is. Including that virtual anatomy lab that Cosumnes River College has put up. It doesn't have the great resources, and our lab tech, Jeff, is supposed to be working on this, but apparently he's incredibly slow. But when you go here, they've got some great digestive system models and charts. And one of the charts you can see here is this great chart for the digestive system. It shows the mouth, it shows the oral cavity, shows a lot of things. But what I wanted to show you in particular here is this view, because what I like about this is it shows us a couple of things. We don't really see the lesser omentum because we see the blood vessels and structures that are in place in front of it. However, if you look closely, there are a couple nice things that you can see. Notice here, coming off of the greater curvature of the stomach, we see some of that greater omentum going down. And notice also they have cut and removed the transverse colon. But what you can clearly see, and I'll highlight it here, is you can see that mesentery that has been cut that would have anchored that transverse colon into place. So that would be the transverse mesocolon. Notice if we work our way through the slides on this, here we see the inferior view of that, where again, we see the greater omentum draping down over the abdominal pelvic cavity. Again, we see that transverse mesocolon really nicely right there. And then we can also see some of that mesentery proper that would hold our small intestine in place. Oh, look, a little bit more of the greater omentum over here as well. So there are these great pictures on this where it does a nice job. Right, there's a nice anus and all sorts of other things, cecum, ileum, liver, mouth, all these things, all these great pictures from the classroom, the charts and things along those lines. There are some good models in here and things along those that you can see as well. So again, make sure you're taking advantage of all of the resources that are available to you when you are learning this anatomy. All righty, now, as I mentioned, this serous membrane doesn't go over all of the organs. Some organs are in the abdominal pelvic cavity or in the peritoneal cavity, and we call them intraperitoneal, whereas others are behind it. Your book's got this simple illustration that does a nice job of showing this. Notice we see things like a piece of small intestine anchored to the back wall by the mesentery. Here we actually see mesenteries coming off the front wall, or really the superior wall, to wrap around the liver and then wrap around the stomach and go. But the point is they are all held by serosas inside the abdominal pelvic cavity. However, some organs, and here we see an example of it right here, even if they start in the abdominal pelvic cavity, during development, they are actually brought behind the abdominal pelvic cavity. And when they're located here behind the abdominal pelvic cavity, as I mentioned, we call them retroperitoneal. And it is the retroperitoneal organs that are covered by an adventitia, whereas those that are intraperitoneal are the ones that are covered by a serosa. So then the big question becomes, what are the retroperitoneal organs? Someone give me an example of one of the retroperitoneal organs. Anyone know any of them? Oh, hold on. Let me answer this question. Okay, so the mesenteries weaken and allow movement of the organs or the greater omentum itself. So again, it isn't so much that the mesenteries weaken, it's that there's a weakening in the wall. You have an increase in pressure in the abdominal pelvic cavity, and that increase in pressure forces the fascicles or forces a weak spot like the umbilicus to expand, and that expansion allows those organs to be forced out. Excellent. Perfect. Okay, so there we've got a couple of them. We've got the pancreas. Now, the pancreas is a feather-shaped organ. This is a little big, so let me switch, get this a little smaller. Pancreas is a feather-shaped organ. The head and body of it is retroperitoneal, but it turns out its tail actually does stick out into the abdominal pelvic cavity. So it's kind of cool that way. We've got this funky organ that is the pancreas, where the head and tail sits behind the peritoneal cavity, but I mean the head and body, but then its tail kind of sticks out into it. But yeah, part of the pancreas does. Hold on, the kidneys absolutely do. Kidneys are retroperitoneal. So the small intestine, yes, but only the beginning part of it. The duodenum or duodenum, both are acceptable. The duodenum of the small intestine is retroperitoneal. In fact, it is what wraps around the head of the pancreas, the duodenum of the small intestine. So again, the duodenum is the part of the small intestine. You are right, the part of the large intestine does as well. And that part of the large intestine is the ascending and descending colons. So again, we haven't fully talked about the anatomy of it yet, but the ascending and descending colon is retroperitoneal, inferior vena cava, and what sits right next to the, there you go, the aorta and the inferior vena cava. Excellent. So what, I'm not sure, the head and the body of the pancreas, yes, head and body of the pancreas is retroperitoneal, the head is not. Excellent. The ureters, excellent, the ureters are, oops. The ureters, excellent, because they come out of the kidney, that makes sense. What sits on top of the kidney? If the ureters that come out of it are adrenal glands, absolutely. But however, remember, the adrenal glands, because they sit on top of the kidney, are also sometimes referred to as the suprarenal glands. So suprarenal glands or adrenal. Excellent. Couple more. Esophagus, excellent. There you go, perfect. And the rectum. Excellent. The third part of the large intestine that is retroperitoneal as well. So there you go. The adrenal gland, the aorta, inferior vena cava, the duodenum of the small intestine, the head and the body, the pancreas, the uterus, the ureters, the ascending and descending colon, the kidneys, the esophagus, and the rectum. These are your retroperitoneal organs. And if you have trouble remembering that, just remember, if you are retroperitoneal, you are a sad pucker. I did say P, pucker, but you're a sad pucker if you are retroperitoneal. All right. Questions on that? All right, excellent. This is a good second stopping point, because at this point now we've talked about all of the basic anatomy, but obviously we have a lot more important material to cover. And so, as I said, I think for our digestive system, the easiest way to work with this is to follow that mystical, magical journey that food takes through the alimentary canal, looking at how it is modified, looking how it is changed, looking at how it is treated as it works its way through the alimentary canal, which means, of course, we are going to start in the oral cavity. So let's go ahead and take our next break. Looks like it is 2:10. So let's come back at 2:25 and we will restart at 2:25. And I will start the recording at that point. All righty. Any questions before we take our next break? All righty. I will see you in 15 minutes. I saw a great question here before, I don't know if it came before the break or during the break, but I wanted to answer it. So the transverse mesocolon connects the transverse colon to the posterior wall of the abdominal pelvic cavity. So not to the rest of the intestines or anything else. As you can see from this illustration here, where we see the entire alimentary canal, basically what happens is that, as we talked about, the ascending and descending colon are retroperitoneal. They are anchored to the abdominal pelvic wall. However, the transverse colon, this part sticks out and so needs to be supported and held in place to the back wall of the abdominal pelvic cavity by a mesentery, and the sigmoid colon also needs to be anchored to the back wall as well. So both of those have mesocolons that hold them in place. The transverse mesocolon holds the transverse colon, and the sigmoid mesocolon holds the sigmoid colon in place. Excellent. So that was a great question. Any others before we dive back into the new material?
I just wanted to make sure. So the mesentery, there's 5 of them for the mesenteries. The mesentery proper is for the small intestine. The sigmoid is for the sigmoid. The lesser omentum is connecting the liver to the stomach. The greater omentum is from the stomach to the transverse colon, and the transverse mesocolon is to the abdominal pelvic wall.
Yeah, I think that sounds right.
Okay.
Yep. And again, you have an anatomy list, but that I know those are on. But yeah, it sounds like there's 5. That sounds right. I don't think you missed any there.
Okay. Thank you.
Excellent. Any other questions? All righty, then let's move forward. Again, our goal is to work our way through the alimentary canal. We just had a 15-minute break, plenty of time to eat 2 or 3 cheeseburgers. So when you first take that cheeseburger and insert it into your alimentary canal, it starts in the oral or buccal cavity. Excellent. So what are the boundaries of the oral or the buccal cavities, right? It's basically a box, depending on how you think of it. And let's just say that this is a sagittal view. So this would be anterior and this would be posterior. What are the boundaries? What are some of the structures that help to form the boundaries of the oral cavity?
Your cheeks.
All right, excellent. So absolutely laterally, we're going to have the cheeks. Of course, again, this cheek has obviously a cutaneous membrane on the outer surface, a mucous membrane on the inner surface, but there is a muscle. And what is the primary muscle that would form the lateral borders of the oral cavity? Buccinator, excellent. That buccinator muscle would help to form the lateral boundaries of the oral cavity. I heard someone say orbicularis oris. Where would that be? That wouldn't be lateral border. What would that be?
Lips.
Anterior. Yeah, absolutely, right? It's that circular muscle that basically forms here, forming the anterior boundary, right? Acts as a valve, allowing it to close, right? So this is the orbicularis oris. That is the muscular boundary, anterior muscular boundary. I think I heard someone, I thought I saw someone say teeth, right? So the teeth would definitely project down. So we have those teeth that would help to form. Let's actually cheat and do this. I'll move, oops, nope, hold on. Back there. I want this. So we're going to be tooth there and tooth here. So those help to form the boundaries, absolutely. I heard someone also mention the lips. Absolutely. So we have an extension of the cutaneous membrane that would come down and help to enclose, wrap around that. And at the edge of it, we have a specialized feature known as the red margin. What we commonly refer to as the lips is that red margin, that thin, hairless skin, glandulous skin that is going to form that margin there. So the lips would be there. There is a space in between the lips and the teeth, right? What would the space between the lips and the teeth be? Anyone know what we call that? Right, if you think back to middle school, right, when you took your first big chaw of chewing tobacco, right? You had to fit in with all the other kids on the badminton team. So they were all chewing tobacco. So you had to do that too. Excellent. That vestibule, that inner region there, spectacular. Excellent. The buccinator by itself isn't going to help to form the lateral boundaries. I saw someone say masseter. That would play a role as well. Would help to form part of that lateral aspect of the cheek. Excellent. So these are lateral borders. So let's cheat and put them inside the oral cavity. Excellent. Superiorly, we have a bony structure. Oops, nope. Superiorly, we have a bony structure that is a skull feature. And that skull feature is called the hard palate. Yep, hold on, we'll get to the soft palate in just a second. Let me, because we got some work to do here first. We have this skull feature that is indeed known as the hard palate. That hard palate I mentioned is a skull feature. Someone remind me again what a skull feature is? What does it mean to be a skull feature? Excellent. Takes 2 or more bones to make it. Luckily, way back in 430, we learned about bones and bone features. So the first question is, how many bones make up the hard palate? That is the bony roof of our mouth. 4, excellent, 4 total bones. And someone give me the 4 bones and bone features that make up the roof of the mouth. Give me one for starters.
Maxillary.
Excellent. Maxilla is definitely one of the bones. What bone feature? And the palatine's the other. Excellent. And someone's already hit on it. These are going to be, there's going to be a right maxilla and a left maxilla. And a right palatine and a left palatine. But we also need to know the bone features of these as well. What bone feature of the maxilla helps to form the roof of the mouth? Excellent. The palatine process, right? Remember, it's a bone feature of the maxilla, but because it articulates with the palatine bone, it's called the palatine process. So therefore, of course, the part of the palatine bone that articulates with the maxilla is called the maxillary process then, right? Wouldn't that make perfect sense? If one's going to be the palatine process, the other one must have a maxillary process? Nope, because anatomists hate us. So what did they call the bone feature of the palatine bones? There you go, horizontal process or horizontal plate. Excellent. Or horizontal process, either are acceptable terms. Excellent. So the bony roof of the mouth is comprised of 4 bone features: the right maxilla, the left maxilla, the right palatine, and the left palatine. And the bone features of the maxilla are the palatine processes. The bone features of the palatine are the horizontal processes. And if we cheat here, because conveniently enough, where am I going? 430 class happens to be in the bones right now. So I can cheat and use one of the pictures from there. I think there's a better picture. Oh, I like that one. There we go. So there we go. So again, here we see that hard palate, that roof of the mouth made up by these 4 individual bones, there and there. So again, this would of course be this would be the palatine process of the right maxilla, palatine process of the left maxilla, horizontal process of the right palatine, a horizontal process of the left palatine. And if I remember correctly, in 430, there's actually a condition where this suture between them doesn't fully form. It can be caused by malnutrition, like a decrease in folic acid in mom. What do they call that condition again? Cleft palate, absolutely. That cleft palate that forms as a result of that. Excellent. There you go. So that's perfect. We've done that. Oh no, I'm going to lost my picture, haven't I? Shucks. All right. So we've done the lateral. check. We've done part of the roof. We've done the front. So that's a good start. So we've talked about the hard palate and what that's comprised of. But I also saw someone mention the soft palate. What is that?
Muscles.
True, although it's not so much muscle. There's a little bit of muscle in it, but it's more primarily connective tissue and mucous membrane that basically forms the inferior part. And thanks to Cardi B, we all know what it becomes at the end. And what is that? Tapers to the uvula. Excellent, right? Of course, that uvula does not form a valve-like structure the same way that the lips and the orbicularis oris do, but it is adjacent to what feature, what organ is posterior to the oral cavity? Pharynx, excellent. That pharynx is a muscular tube that is the posterior part that it is continuous with. Excellent. So I think really what we're left with is what is the inferior boundary of the oral cavity? Tongue. Excellent. Obviously, the tongue can move up into the oral cavity. The mandible is part of the bony feature. I would give it that. But again, the trick with the tongue is the tongue, basically the muscular to it, remember, starts out and it can project into the oral cavity, but it is also anchored to connective tissue and that movable bone. What is that movable bone that is the base of the tongue? Hyoid bone, excellent, right? In fact, if we instead go back here, this is what we want. Let's switch to this picture. Notice here we can see some of the things that we've been talking about. Here is that hard palate. Here is that soft palate that dangles down. Here are the labia, the lips with the red margin, the teeth, and that space in between, as we mentioned, is the vestibule. Right over the lateral wall, we can see the buccinator and the masseter, which would form that boundary. But notice here is the tongue. While the tongue can project into the oral cavity, right, you can see, I guess maybe you could argue the mandible is a little bit of it as well. And then that right there is that hyoid bone, that movable base for the tongue. So I'd say the tongue would be fine. If you wanted to say the mandible, I would say sure. Just make sure you include the tongue in that. And then, like I said, it is continuous with the pharynx, which is this entire muscular tube that is divided into 3 distinct regions based on the openings. And so this would be the oral pharynx that it would open to there. So we see these nice boundaries. We see it nice here. I think there's one other picture I wanted to show you. When we did that. We'll come to all of those. But I thought there was the front view. No, okay, I have the front view. That's what it is. So there we go, this view as well. Excellent. All right. Now, remember we said from the distal third of the alimentary canal down, it is simple columnar epithelial tissue, but not in the mouth, not in the pharynx. What is the tissue type that lines our oral cavity?
Non-keratinized stratified
There you go. Non-keratinized, stratified, squamous epithelial tissue or epithelium. Perfect. Make that bigger. Excellent. So we have that. And again, it is non-keratinized. If we were ever getting back into the classroom again, one of the fun activities we can sometimes do in there is do a little cheek scrape, put it on a slide, stain it, and see those nuclei that are in the superficial layer of that. However, and again, we are all locked up, so you're not able to do this. If we were in the classroom, I would tell you to grab the person next to you in the classroom, pry their mouth open and look inside. Feel free to do that to anybody that you are quarantining with at home. They won't mind. And when you look inside their oral cavity, one of the things you may notice on the roof of their mouth or on the surface of the tongue or along some of the gums, you might see some whitish coloration to those areas. And the reason for that is while the oral cavity is primarily non-keratinized stratified squamous epithelial tissue, the same way that if you do a lot of abrasion on your skin, calluses can form where the cells attach and harden in that. It's the same thing in the oral cavity. In the oral cavity, you can actually get some minor keratinization if you're eating a lot of spicy food or a little hot food or damaging it in some other ways. Maybe you're brushing too vigorously. You can actually slightly keratinize certain regions of the oral cavity, and that'll be expressed as a whitish coloration to the tissue inside that oral cavity. All right. This is a lot of the anatomy, although there is some more that we need to be able to talk about. Again, we have talked about the labia. There are two, the superior and the inferior. And again, I want to make sure we emphasize that when we talk about the labia, I'm talking about everything from the nose down. That is technically the labia. Often when we say the word lips, what we think of is just the edges. And as I mentioned before, those edges are a region known as the red margin. This red margin contains a large number of sensory receptors. It is relatively thin skin, so it is a very thin layer of keratinized. It's actually the transition area from the keratinized outer surface to the non-keratinized inner surface. And as such, it is hairless and it is glandless. Right? Hairless is good because you don't want hairs sticking off the edge of your lips. That would be gross, right? Wouldn't be a lot of fun. It'd tickle all the time. But there's a problem with it being glandless. Right? It can't lubricate themselves. So how are you left to lubricate your red margins to maintain the moisture so they don't crack?
Smile.
No, smiling doesn't necessarily lubricate them. Yeah, you got to lick them, right? Or at least put your bliss sticks or something like that on there, your Burt's Bees, right? To maintain the moisture of those, exactly, because they cannot be moistened. Otherwise, we have to self-moisten the red margin for us. Right. Again, here with these retractors, we can see into the vestibules. And notice one other thing, despite what our president seems to be showing us, our lips don't truly flap around aimlessly, right? Instead, they are anchored in place by these extensions of the mucous membrane. And this folded extension of our mucous membrane is what is called a frenulum. You have two labia, you have two vestibules, you have two frenulum. So you have the superior labia with the superior vestibule and the superior labial frenulum, and the inferior labia, inferior vestibule, and inferior labial frenulum. In fact, you have three frenulums, 1, 2, and 3, one for the tongue and 2 for the labia, which is why we need to specify labial frenulums versus the lingual frenulum, which we'll talk about in a bit. We did the oral cavity and the things along associated with that, but there are two other features that I want to show you here. This view, we talked about these before in the last section, but I want to make sure to emphasize them here. Remember before we were talking about the tonsils, the palatine tonsils, and how the palatine tonsils are located between two arches. Notice one of these arches goes from the roof of the mouth down towards the tongue. This is called the palatoglossal arch. Notice the second one goes from the roof of the mouth down towards the pharynx. So it is the palatopharyngeal arch. And then in between them is an indented space. That indented space is known as the fosse. And that indented space, that fosse, is where you find the tonsils. Remember, we saw this once before when we were looking at that chart. Remember, this chart showed this great view here. So notice with this great view here, again, we have, this is that palatoglossal arch where we can see it. We don't obviously see it connecting to the hard palate because it's been removed, but we can see how it projects down towards the tongue. And here we see that palatopharyngeal arch where it's going back towards the muscular pharynx that we see here. The indented space in between those arches would be the fosse. And at this particular level where the cross section has been made, we can see that that palatine tonsil is located within that fosse. So again, this is not new anatomy. We talked about it in the last one, but we see it nicely here as well. All right. Questions on that? Excellent. We did all that as well. All right. So I think that is the basic anatomy of the oral cavity. I think we have done an excellent job of beating that to death. So let us talk now about the functions. What are the functions that occur in the oral cavity?
Mechanical.
Excellent. So it's perfect. Mastication. Mastication is what we commonly refer to as chewing. And of those 6 or 7, depending on how you want to think of it, functions of the digestive system, which one does mastication do?
Mechanical digestion.
Excellent. So this is mechanical digestion, right? Taking big pieces of cheeseburger and breaking them down into little pieces of cheeseburger. I saw someone put ingestion. In fact, this is the only location where ingestion occurs. Excellent. Right, that is that forcible shoving of the cheeseburger into the mouth. Is there chemical digestion that takes place?
Yeah.
Yeah, absolutely. Right. Excellent. In our salivary glands, we produce a special enzyme called salivary amylase. And what does that salivary amylase do?
Starts digestion of starch.
Excellent. This digests starch. It digests the simple sugars. You can actually see this for the cells. Yeah, very simple digest. I like that better. Carbohydrates. Because again, our 4 main macromolecules. Someone remind me again of what our 4 main macromolecules are.
Protein, carbohydrates, lipids.
Carbohydrates, proteins, I heard lipids, and what's the 4th? There we go, nucleic acids. Excellent. So not alcohols, nucleic acids. So those are our 4 macromolecules. And notice the breakdown of carbohydrates starts right away in the mouth. Again, we don't have some of the labs that we can do, but there are plenty of fun things you can do at home. And one of those things that I encourage you to do at home, grab you and your significant loved ones that are around the table, take a paper towel, dry off the surface of your tongue, and take a little cube of white bread. And take that little cube of white bread and put it on the surface of your tongue and just let it sit there. And as you just let it sit there, what will happen is your tongue will start to produce saliva, your salivary glands will start to produce saliva. It will mix with that bread, and you will start to taste a sweetness on the surface of your tongue from the breakdown of those carbohydrates into simple sugars. Absolutely. What else? Absolutely, excellent. So what do you mean by sensory? Taste, absolutely. So taste. Excellent. We have taste buds, special chemoreceptors that allow us to perceive our sense of taste. How many different taste sensations are there?
5.
Excellent. 5 basic taste sensations. What are they? All right, I saw sweet, salty, umami, right? That
Sour.
savory sense, sour, and Now we'll talk about pain in a minute. Pain isn't a taste sensation, but bitter, excellent. Bitter, that is definitely a taste sensation, absolutely. So those are them. We can feel things like pain and temperature, but that pain and temperature sensation is not taste, right? Those are more of like tactile type sensations. Right, so we're getting tactile sensations like that. Notice spicy is not a taste either. Well, again, remember all of these taste sensations are chemoreceptions. So basically you have a chemical that binds to a receptor like a key and turns that lock. And when it turns that lock, then what ends up happening is you perceive that taste sensation. Right? And we have 5 of them: sweet, salty, umami, sour, and bitter. Spicy, things like capsaicin. Capsaicin is that janitor key. And what does a janitor key open?
Water.
Everything. Exactly. And that's what happens. That capsaicin triggers all of these taste receptors, right? And even the pain receptor as well. And so you get this big, huge, confusing signal sent to the brain that is perceived as painful, right? It also tricks the temperature receptors to make you think that your mouth is really hot. So you start to get red in the face, you start to sweat, right? You get all those reactions from that spicy taste sensation because it is overwhelming both the sensory and the tactile sensations in your oral cavity. Excellent. Right? What else? What else do we do in our oral cavity? We got chemical digestion, right? Again, as we talked about, that chemical digestion is part of a secretion. So if we wanted to think of that as a separate function, what is secreted in the oral cavity?
Saliva.
Saliva. Excellent. So we have glands that produce that. That contains things, lots of things. Those defensins that we talked about, lysozymes that we talked about when we were talking about our body defenses, salivary amylase. Notice also there's a lingual lipase as well that is released into the oral cavity. What do you think a lingual lipase does? Well, we'll talk about speech in a minute. You're right. Speech is something else that happens in the oral cavity. It's not a digestive function. I would say that's a non-digestive function, but it's an important one. What about lingual lipase breaks down lipids? Absolutely. So does that mean that if I took a chunk of fat and put it on my tongue, it would start to break down on its own and I'd be able to taste the fatty acids and alcohols that are coming off of that?
No.
No, because it's actually produced, when it's produced in an inactive form. It's produced in an inactive form. It doesn't actually become active till it reaches the stomach. Why is that? What would cause it to become active in the stomach if it's not active in the oral cavity?
Acid.
There we go. The pH change, absolutely, the acid in the stomach. What's the pH in your oral cavity?
Basic.
Yeah, well, it's about 7. Yeah, pretty neutral, right? What's the pH in your stomach? Yeah, 1 to 2, very, very acidic, absolutely. As we know, proteins are very sensitive to pH and temperature changes. So when the pH changes, it is converted into an active form and can start the breakdown of lipids. So notice lipid breakdown or chemical breakdown of it anyway, starts in the stomach, carbohydrates in the oral cavity. What else? So let's see, we had chemical digestion, we had mechanical digestion, we've got ingestion, we've got secretion. Does absorption occur in the mouth?
No.
Yeah, it's limited, but it does absolutely occur. There are some water, for instance, can be absorbed in the oral cavity, certain medications, right? When grandpa's having one of his heart episodes, what is it that you want to do to grandpa? If Grandpa's having heart problems, right? And then he turns on the Senate debates and gets all worked up and starts to have heart issues. What do you have to do to Grandpa? I mean, obviously you turn the TV to the Game Show Network, but what else do you do? Come on, what do you do with people who have heart problems? I know you guys know this. I will wait. There you go, a nitro pill. And where do you put that nitro pill?
Under tongue.
Under the tongue, absolutely, excellent. Right, so certain medications can be absorbed. So certain fatty acids can be absorbed. So absolutely, sublingual, under the tongue, exactly. So there are some absorption that can take place there. Excellent. So now we have chemical, mechanical, absorption, secretion, mastication. What were the other two left? There's only two more. What are they? Propulsion. Is the oral cavity responsible for propulsion? Yeah, absolutely. What do we call the process of propulsing food out of the oral cavity along its merry way through the alimentary canal? Swallowing, absolutely, right? We'll commonly refer to it as swallowing, right? Or we can use the appropriate anatomical term, deglutition. Deglutition is the act of swallowing food down. Notice all of the functions of the alimentary canal, with the exception of defecation, take place in the oral cavity. And thankfully, defecation does not occur in the oral cavity. But all the other ones do, including other functions like sensory, like tactile sensation, non-digestive functions like speech, right? There's a whole lot going on inside of our oral cavity.
Also,
All right.
also
I'm sorry?
Also, we can breathe via mouth.
True, technically air can move through that way, but remember, the oral cavity is not part of the respiratory system.
Yeah.
The same way I could put a straw up my nose and get milk into my stomach that way, but that doesn't make my nose part of the digestive system. Anyone who's had a cold survived it because they were able to move air through their oral cavity, but it's not a part of the respiratory system.
I had a question.
I have an answer.
Would it be okay if we wrote swallowing instead of deglutation? Well, if you asked about swallowing, or do you want?
Well, but so think of it this way. What do you think is more likely to happen? Am I going to ask a question where the answer to that question is either going to be swallowing or deglutition? Or is it more likely that I'm going to ask you to describe the process of deglutition, and you need to know what deglutition means so you can describe the process?
Okay, makes sense.
Yeah. Now, that being said, if the way that I could see that question, like as a fill in the blank question, would be, what is the anatomical term for swallowing or something like that? I guess technically I could ask, what is the common term for deglutition? So I could see that maybe as a fill in the blank type question, because it's vocabulary and that's kind of what the fill in the blank are. So it's probably not a bad idea to know that term. And like I said, we will actually talk about the process as well. All righty. Excellent. Now, the oral cavity is also the location of several accessory organs, starting first with the tongue. What is the function of the tongue?
Doesn't it help with like forming the bolus?
Excellent. More fancy terms. I like that. Absolutely. We got mastication. We have deglutition, right? We now have bolus. Absolutely, right? Food, right? That cheeseburger undergoes iterations. It undergoes changes as it works through the alimentary canal. And that first change, that first iteration that occurs in the oral cavity, right? If you're at a meal with a loved one and they feel like sharing their cheeseburger with you and take out a knife and cut it in half, you have no problem with that. If they put it in their mouth first and chew it 3 or 4 times and then spit it on the plate, are you as likely to want to eat that? It'd have to be a really good cheeseburger to want to do that, right? Because inside the oral cavity, with that secretion, with that mastication, absolutely, our food undergoes its first iteration, its first change, becomes a big gloopy mess that, as was mentioned, called a bolus. So yes, moving that food around so that it can be presented to the teeth for mastication, producing secretions that are going to help to mix with that food to form that bolus. Absolutely, those are functions of the tongue. And what else does the tongue do for us? Taste, perfect, and produce speech, and plays a role in that deglutition process as well. Absolutely, excellent. So there's a lot of important functions here in the oral cavity. Notice it has a very unique and distinct shape to it. There are several different prominent features that stick out. But one of the first things that should be noticed about it is that there is primarily a major border distinguishing line about 2/3 of the way along the length of the tongue. We have the anterior 2/3 of the tongue and the posterior third. They're very different in structure, and not surprisingly, they're very different in function. Notice the posterior 2/3 is the part that we've kind of already talked about. This is the part that is formed where all the lingual tonsils are located. Plays a much more important role in our immune response, right? Those are where those lingual tonsils, whereas the anterior 2/3 does most of those functions we just talked about, moving the food, producing speech, taste, all those things that we talked about as well. And that boundary between the two is what is known as the terminal sulcus. When we come anterior to the terminal sulcus, the tongue does not have a smooth surface on it. There are basically 4 primary types of bumps, finger-like extensions, or what we call papillae, that stick out. The largest of these are what are known as the circumvallate papillae, or what are also sometimes now referred to simply as vallate papillae, because apparently circumvallate was too hard for people to spell. They form a large distinct line just proximal to that terminal sulcus, and they house taste buds. They're one of the major locations where there are a large number of taste buds that help us to perceive the chemical composition of our food. Over the rest of the surface of the tongue, we see the distribution of these medium-sized papillae. And these medium-sized papillae are what are known as the fungiform papillae. And these fungiform papillae also house taste receptors in them, those taste buds. And we'll take a look at those histologically in just a minute. How are we doing on time? We're doing great. Excellent. The third type of papillae are these ridges on the lateral aspect of the tongue. These ridges kind of make it look like the edge of a leaf, like foliage, which is where they get the name as the foliate papillae. What's interesting about these foliate papillae is that they house taste buds in an adolescence or in an infant. Many of the taste buds here happen to be specialized for milk fats and milk sugars. Why might having taste buds for milk fats and milk sugars in an infant be useful? Not your question. What do infants eat? Milk. There you go. Exactly. However, as we age, those go away. And one of the theories about these foliate papillae is that if you notice, many children tend to be much more sensitive to food taste than adults are. Some of them can be incredibly picky about the things that they will eat, right? They would rather have their left arm ripped off than eat a mushroom, whereas you barely taste a mushroom and don't mind them at all. And there's some speculation that it's these extra taste receptors in the foliate papillae that make kids more sensitive to certain tastes, like broccoli, like mushrooms, like things along those lines, which is why they have much more of an adverse reaction to those things than some other individuals may have. But notice that's only 3. There is a 4th type of papillae, and this illustration really doesn't show it. Basically, anywhere on the tongue where it looks clear in color or smooth in its appearance, it isn't. There are these tiny little hair-like papillae known as filiform papillae. These filiform papillae, unlike the other 3, don't and never contain taste buds. So then what do? If they don't house taste buds, well, part of it, not so much sucking, although the sucking is obviously forming that seal with the entire oral cavity. Latching, I'm not sure I would go with that as well, but you do have the right idea, friction. Right? How many people have ever seen a cat drink milk before? Or water, right? When a cat drink. Does a cat pick up the bowl and take a big swig of it? Does it scoop it with its paw? You get the right idea. It scoops it, but it doesn't scoops it with its tongue. Exactly. Basically, it dips its tongue in. The tongue of a cat have very pronounced filiform papillae that give it friction. So much friction, it's able to use those prominent filiform papillae to be able to pick up water. And for the food as well. Now, I know your kitty gets that wonderful cat food, right? That you do it in the can and scoop it into the crystal bowl and all those types of things. But again, if you think of a, feral cat, usually it's eating something that doesn't want to be eaten. And so having a tongue that has a tremendous amount of friction on it helps it to be able to hold on to its prey and get it all the way into its mouth. All right, anyone here been licked by a cat before? Couple of you, anyone? Yeah, there you go. A couple of people willing to admit it. Absolutely, that tongue has a very rough texture to it, right? How many people here have been licked by a loved one? Did it feel the same as being licked by the cat? No, your significant other's tongue feels very, very different from a cat tongue, right? Because our filiform papillae are not nearly as pronounced. They can still provide some some friction, right? Although again, unlike that cat that's eating something that doesn't want to be eaten, our diet, of course, includes anything you can buy through a drive-through. So is there any other function of these filiform papillae? Well, let me ask you this question. If you had a marble and it was clean, well, the muscles change the shape of the tongue. We'll talk about that. If you had a clean marble and you put it in your mouth on the surface of your tongue, would you know that it was there?
Yes.
Yeah. You would be able to tell that it was smooth, right? Or if I put a block, you'd be able to tell that it was rough or its shape, right? If I heated it up in the oven before I put it in your mouth on your tongue, would you know that? Yeah, remember we talked about tactile sensation and temperature sensation. That's what the filiform provide for us as well. They provide friction, tactile sensation, and temperature sensation. And we have these all over the surface of the tongue. I think I have a couple good pictures here. We did that. We'll talk about those in a second. Let's look at the pictures first. Here we see that circumvallate papillae, that big, large papillae that is going to house a large number of taste buds. Here we see at the center of this, we see a fungiform, mushroom-capped name. That's where it gets its fungiform. And then all these tiny little quill-like, pointy-like papillae are the filiform papillae that surround them. Notice when we look at these things histologically, and we see a nice view of this here. Here we see the pointy filiform papillae on the surface here. Here again are those largest, the circumvallate. And on this large circumvallate, here on the lateral edges, and also this particular section doesn't show it, but the circumvallate papillae can also have taste buds located on the anterior surface as well. This is different from the fungiform and the foliate, which again, histologically look very, very similar to each other. You don't have to worry about distinguishing these two. But their taste buds are only on the lateral aspect. And I wanted to give you an up close view of this taste buds as well. So again, here we can see on the lateral aspect, these circular cup-shaped structures are the taste buds. These taste buds, and let me see if I can find a good one. Interesting. None of these. I guess this is the closest one. So sometimes with the plane of section, what you can actually see when they cut them, you get lucky, you can actually see the pore, the little opening where the saliva and the chemicals that are dissolved in that saliva are able to get into that taste bud and be able to perceive the taste. And that is the key. We perceive taste by mixing that chemical with our saliva. Gustatory neuroanatomists are a very odd bunch, but they do some interesting research. And one of the things they were curious about was how precise our taste sensations as humans can be. And one of the things that they found is that people were much more precise at being able to detect a taste sensation when it was dissolved in their own saliva, as opposed to just dissolving it in distilled water or dissolving it in someone else's saliva. So if you think of what these studies are like, basically what happens, you as a subject get to sit there and they have 3 vials. In those 3 vials, they have some of your saliva, they have water, and they have someone else's saliva. And they put a little bit of chemical into each of them, dry off your tongue with a paper towel, stick a Q-tip in there, and rub it over the surface of your tongue. And first they do that with your saliva, then they rinse your mouth, then they do it with the water, then they do it with someone else's saliva. That is a great question I don't have the answer to. And what they found is that people are much more sensitive to things in our own saliva. So it's that dissolving of the chemical in our saliva that we're able to perceive. I know, so here's what I would say. COVID somehow affects our ability to smell. For many people, one of the symptoms associated with COVID is a loss of sense of smell. Now, smell and taste are both chemoreception, and the two are very much mixed together. I'm not hoping anybody gets COVID and anybody experiences this, but the next time you get a cold, one of the things that you can do, or better yet, do it to somebody else who has a cold. If your significant other has a cold, and it's just a cold, but they're very congested in their nose, what you can do them is give them a thin slice of onion and a thin slice of apple and have them eat them. And typically they can't tell the difference between the two. The reason for that is our sense of taste is very much influenced and affected by our sense of smell. Olfactory neuroscientists are even weirder than the gustatory ones. And what's one of the really interesting things they found is not only is it the sense of smell, the chemicals that are absorbed in the moisture of the air, but also the moisture of our saliva as well. One of the things that they have found is that the sense of smell you get from a chemical when it wafts up the posterior part through your pharynx into the nasal cavity, as opposed to from outside, you perceive it differently. Where they see this a lot is in a lot of funky cheeses, right? A lot of people, when you come up and you first smell, one of those really prominent funky cheeses, it outside smells very, very bad. But when you taste it, it tends to have a much more pleasing taste to it. And that's because you're smelling it from the inside as opposed to from the outside. And they found differences in that. All right. Excellent. So back to here, because I don't want to go, hold on, clear that. Go here. Lastly, as I mentioned, our tongue has many muscles in them. Some of the muscles, and I'll just do this in black because it's simple. Some of the muscles are completely contained within the tongue in some type of orientation, and some start outside the tongue and come into the tongue, or start at the hyoid and come into the tongue. Muscles that are completely contained within the tongue are what we call intrinsic muscles, and muscles that start outside the tongue and enter into the tongue are called extrinsic muscles. And not surprisingly, intrinsic and extrinsic muscles do two different things. What do extrinsic muscles do? Anyone? Yeah, extrinsic muscles are move the tongue through space. So you stick the tongue up, you put it down, right? Move it around. That ability to move your tongue in space is what the extrinsic muscles do. So then what do the intrinsic muscles do? They change the shape of the tongue. Exactly. Rolling your tongue, flipping your tongue over. Some people can put ripples along the surface of that tongue. That ability to change the shape of the tongue is what your intrinsic muscles do. And what controls both? Muscles of the tongue, excellent. Cranial nerve 12, the hypoglossal nerve. Excellent. Inferiorly, we've talked about the dorsal surface, but inferiorly underneath it, the plantar surface of it, again, there is a fold in the mucous membrane that anchors the tongue, limiting its range of motion. This is a good thing, except when it's not. As someone mentioned earlier, if the lingual frenulum is too short, then there can be a problem with latching on, right? The baby can have a hard time forming suction to be able to express breast milk from mom's breast. It can impact the ability to swallow. It can affect the ability to produce speech, right? Exactly. The common term from that is absolutely being tongue-tied. However, are you going to be able to get away with a term like tongue-tied on this exam? No, of course not. You are going to need the appropriate anatomical term. And anybody know what that appropriate anatomical term is? Not tongue-tied. That would be nice. Nope. Ankyloglossia, which you know is a term you're going to have to spell on the exam at some point. Right? There you go. Ankyloglossia. Now, how do they resolve it if someone is tongue-tied, if someone's lingual frenulum is too short? Exactly, they cut it. They basically make an incision in it, cut it, and that loosens the tongue, allows more free range of motion. Right? Of course, it makes the tongue a little bit looser in the mouth and more easy to move around. Can give you potentially an extra long tongue. It has never been proven, but there's a tremendous amount of speculation that Gene Simmons, who famously from Kiss is known for having an extremely long tongue, that he either purposely had it cut or actually tore it. And that is what allows him to extend his tongue so much more prominently as a result of that. And like I said, because tongue's not doing enough, remember it also produces that lingual lip base. All right, questions on that? All right, hold on a second. All right, that'll work. All righty. So that is our first accessory organ associated with the tongue, with the mouth, the oral cavity. Any questions on that? All right. What we are left with, and the last thing we will talk about for today, is our second set of accessory organs associated with the oral cavity, and these are the salivary glands. We talked about these salivary glands way back in 430 when we were talking about our autonomic nervous system, and we were talking about our parasympathetic pathways that lead to 3 pairs of salivary glands. Now we'll actually get to talk about them and talk about what innervates them and controls them and all the fun things that go along with that. But what we have is a total of 6 salivary glands, 3 paired glands, so 3 glands on each side. The largest of them is the parotid gland. It is also the most superficial. That is this one here. Notice it is just inferior to the zygomatic arch, just anterior to the ear, and sits on top of the masseter and the buccinator. We have had the sublingual, which is the smallest of them, and it is located immediately inferior to the tongue, as its name would indicate. And the third one, which is the deepest or most inferior of them, deep in the socket of the mandible, is called the submandibular. These are our glands, and each of them have ducts that are going to allow them to express their secretions into the oral cavity, because after all, these are all exocrine glands. Exocrine glands, of course, have 2 components, secretory structures and ducts, and they express with those ducts outside of the body, in this case, into the oral cavity. Let's look at all 3 of these, starting first with the parotid gland. As I mentioned, it is the largest, just inferior to the zygomatic arch. And it primarily produces serous fluid. Let's talk a little bit about saliva in a minute, but we can do it quickly here. Saliva basically has 2 components. And those 2 components that it has are, first, serous fluid. What is serous fluid usually used for?
Lubrication.
Lubrication, absolutely, right? If you were to rub your hands together right now, they would start to get hot. But if you spit on them first and then rubbed them together, would they get hot right away? No, because that serous fluid would reduce the friction. That serous fluid is also going to contain the enzymes, the defensive molecules, the antibodies, lots of other stuff in it as well. But remember, serous fluid is a very watery secretion, right? And as an older sibling, and for all of those of you who are older siblings as well, you know you are required by law, even still now, as a 40-whatever-year-old, every once a month, I have to kneel on the chest of my younger sibling and dribble just the teeniest bit of saliva out of my mouth so that it almost touches her nose, and then right before it touches my younger sister's nose, I am able to suck it back up. Why does it have that cohesion to it? Because saliva isn't just serous fluid, but it also contains mucus. Of course, mucus isn't what the glands make. What do the glands make? What's the protein that is produced that becomes mucus? Mucin, excellent. Mucin is that protein that when hydrated becomes mucus. So saliva has 2 components. However, what's interesting is our parotid gland primarily just produces serous fluid. So of the 2 things that could be made in the parotid gland, we're really only making one, serous fluid. Again, it has the salivary amylase. It has all sorts of other secretions to it. And notice one other thing about the parotid gland. It has the longest duct. This duct, if you notice, goes over the masseter, penetrates through the buccinator, and then enters into the vestibule, the superior vestibule. Let's be specific about that. the superior vestibule of the oral cavity, right at the level of the second molar. So they come in right to the side. Notice it makes sense that it would produce more of a serious solution. With such a long duct, would you want to try to shove mucus through something long and narrow like that? Not very easily. So having a very watery secretion, it makes it a little bit more convenient to just have one long, narrow duct to be able to release this. Now, we obviously need to be able to recognize the parotid gland from a model or a chart or an illustration like this. This picture shows it nicely. There's a great picture in one of those charts. I'm not sure if it's this one or not. No, that's not what I want. That's not what I want either. Oh, there it is. Here we go. This one's a nice one. So here we see, again, there's the parotid gland. Here we see that parotid duct going over the masseter and through the buccinator into the vestibule here as well. So we can see that really nicely. So obviously we need to be able to identify these things on models and charts and things along those lines, but we also need to be able to recognize our glands histologically as well. Of course, when we're identifying things histologically, we of course need to obviously be able to identify the gland. We need to be able to identify the substance it produces. But also, as we know from 430, anytime we talk about a gland, we also need to be able to identify its functional classification and its structural classification. All right, so those are the 4 things we need to be able to do. And let's go ahead and cheat. Make a white box up here that I can then hopefully move that on top of. Nope, I didn't think that was gonna work. Yeah, because I made the box first. All right, so we'll move the box here and then we'll rewrite it. We want to be able to identify the gland. We want to be able to identify the substance it produces. We want to be able to identify its functional classification. And we need to be able to identify its structural classification. Excellent. Those are the things we want to be able to do. So we can do that. We can do that. We can do that. Perfect. And then I can erase this. Excellent. Let's start easy. What gland is this? Parotid. Parotid, because there we've only talked about one so far. Parotid salivary gland. Excellent. And what is the substance we said it is going to produce? True, saliva, you'd get your partial credit on the exam, but we want to be more specific than that. Remember, saliva has 2 components, serous component and the mucin component. Which one does our parotid gland did we say it produces? Serous fluid. So this one mostly, so we can say mostly. Serous fluid. Excellent. And let's start with one more easy one. Apparently I can't spell functional, but we can hopefully figure out what the functional classification of this is. And if I remember back to 430, how many different functional classifications? Well, first of all, remind me again what functional classification means.
Their functions, how they release its content.
Perfect. Exactly. How does this gland release its contents? Excellent. And how many functional classifications in the whole wide world are there?
3.
3. Excellent. 3 in the whole wide world. How many of those do humans have inside of them?
2.
2. What are they?
Merocrine and holocrine.
Excellent. Merocrine and holocrine. Merocrine mode of secretion is where the cell does what? What is the merocrine mode of secretion?
It's they release their contents via exocytosis.
Excellent. It releases by exocytosis. What is holocrine mode of secretion? Yeah, cell accumulates the contents and then ruptures and releases it. Excellent. Salivary glands, which one does it use? Merocrine or holocrine? Merocrine, excellent. Perfect. So we have answered 3 of our 4 questions so far. And that brings us to our 4th, structural classification. Now, when we talk about the structural classification of a gland, when we're talking about exocrine glands, remember exocrine glands have 2 parts. They have ducts and they have secretory structures. The secretory structures are what make the secretion and release the secretion, and the ducts are basically just the PVC pipe that carries it and releases it out of the body. Do we see both those things here on this histology slide? Do we see ducts and secretory structures?
We see duct. I see the duct.
Excellent, right? Absolutely. All right, so what is this structure right here? Duct. What is this structure right here? Duct. What is this structure right here?
Duct.
What is this structure here?
Duct.
Goose. Duck, duck, goose. Never mind. All right. I laughed. All right, that's all that matters. Absolutely. We have ducts. We see the ducts. Excellent. So that means everything else that we see in the field of view here are the secretory structures, the things that are making the secretion. And I noticed this is a ball-shaped, dark stained secretory structure. This is a ball-shaped, dark stained secretory structure. This is a ball-shaped, dark stained, ball-shaped, dark stained, ball-shaped, dark stained, ball-shaped, dark stained, bar-shaped. Everywhere we look, we see dark stained, ball-shaped secretory structures. Now, ball shape, that means something. What were the ball-shaped secretory structures called?
Alveolar.
Alveoli, excellent. Or acini, you can also use the term acini. Either of those are acceptable terms. Excellent. So we have found our secretory structures. We have found our ducts. So when it comes to figuring out the structural classification, we need to count the number of ducts, we need to count the number of secretory structures, and that tells us our structural classification. If we have 1 duct and 1 secretory structure, what type of gland is that?
Simple.
1 duct, 2 or more secretory structures, what type of gland is that?
Branched.
2 or more secretory structures, 2 or more ducts, what kind of gland is that?
Compound.
Compound. How many ducts do you see here?
1.
Really?
Oh, you mean it's 4?
I
Okay.
counted 4, but do
Yeah.
I really even need to get up to 4?
No, it just has to be more than
Yeah,
one.
once you get 2, you're pretty much there. And how many secretory structures are there here? Anyone want to count them all?
More than one.
More than 1. That's really all we need to know. More than 1 duct, more than 1 secretory structure. So that tells us that this gland structural classification is what?
Compound.
Compound. And ball shape or tube shaped secretory structures.
Ball shape.
Ball
Oh,
shape.
it's
So compound alveoli, or our adjective, or compound acinar. Either of those would be acceptable. So our structural classification is a compound alveolar gland. Now, I want you to notice one more thing. On the exam, I could very easily show you a picture that looks just like this. All right. Of those 4 questions, the easiest one to answer is the last one. When you look at this, you see multiple ducts, you see multiple ball-shaped secretory structures, and you know this is a compound alveolar gland. But notice, as I mentioned, there is a second key part to these acini. These acini are very dark stained. What are the 2 parts to saliva again?
Mucus and serous fluid. Mucus.
Mucus and serous fluid. We've seen many cells that make mucin. And when we look at a cell that makes mucin, when it's filled with mucin on the inside, what does it color does it tend to be under the microscope? White or clear. Do these cells look white or clear? No, they look very dark. So that tells us these are definitely not making mucin. And if they're not making mucin, they must be making serous fluid. Now, I said compound alveolar was the easiest question, but really the easiest question is this one right here, because guess what? All of our salivary glands produce saliva using the merocrine mode of secretion. So it doesn't matter which salivary gland I show you, your functional classification is always going to be merocrine. So that makes it easy. Oops, oh no, didn't want to do that. Okay. So we know it's merocrine mode of secretion. We can figure out it's a compound alveolar. We see that all of these alveoli are making serous fluid. And so we know the one salivary gland that just makes serous fluid is the parotid salivary gland. And so when I look at this slide I can use to answer all of those questions that we just mentioned. Compare this to this. Notice we have another gland. I'm going to cheat, grab that, move it out of the way, which should allow me to erase that and that and that. Oops, nope. Perfect. And then I should be able to put that back. Excellent. Guess what? We have a second salivary gland here. We know it uses the merocrine mode of secretion. Bingo, that's easy. Do we see the 2 components that we want to be looking for? What are the 2 things we need to be looking for again?
Ducts and the shape.
Yeah, ducts and secretory structures. Excellent. Do I see a duct?
Yes.
Do I see a duct?
Yes.
Do I need to keep looking?
No, because it's already more than one.
It's already more than one. And if I know it's more than one, then what does that tell me my structural classification is?
Compound.
Compound. Excellent. That's a good starting point. Now I need to look at my secretory structures. Notice when I look at my secretory structures, some of them are circular in shape, but are all of them circular in shape?
No.
No. Notice this one here is very elongated. This one here, I don't know what shape this is, pretty funky. Here's a pretty funky curved shape to them. Here's more longitudinal ones, right? When you have an acini, when you have a ball-shaped structure, does it matter how you cut it? No matter how you cut that ball, it's gonna be ball-shaped. What was the other shape of a secretory structure? It's gotten dark in here. Hold on a second. Ugh. I feel like I'm going to cave in my mother's basement or something. All right, that's better. Tubes. Now, with a tube, is it possible to cut it to make it look ball shape? Yeah, but is it also possible to cut it to make it look long or cut it at an oblique angle and get ovals? right? Or have it bent and see curves. There's all these different irregular shape secretory structures. So if I'm seeing non-uniform secretory structures, are they likely to be ball shape? Or are they likely to be tube shape?
Tube shape.
Tube shape. So what is the full structural classification of this gland?
Compound tubular.
Tubular, excellent. It is a compound tubular gland. Notice one other thing about these tubes. As far as the eye I see nothing but tubes. And are they really dark stained like that last slide we looked at?
No.
No, these are very light in color. Not quite as light as like adipocytes, right? Adipocytes are completely clear, right? There's little chunks of stuff inside of these, and the nucleus can be in the middle of these, but they're very light in color, which tends to tell me when I see things that are very light in color, what is the substance that is being produced here? If they're light in color, are they making serous fluid? No, they're making mucin. Excellent. We are making mucin here, which of course, when it hydrates, is going to become mucus. So notice, just looking at this histologically, we were able to work through the information and figure out its structural and its substance that it is producing. And now I will tell you that the salivary gland that just produces mucin is the sublingual. So when you see this histology slide like this, you will know without a shadow of a doubt, you are looking at the sublingual gland. Let's go back now that we've looked at the histology and look at the information for this one. So I need to clear that. Oh, I didn't want to do that. I want to move. Screw it, I'll clear it all. Excellent. So sublingual gland, remembers this one underneath the tongue, under the floor, under the mucous membrane. It primarily produces mucus. There's also some buffer, bicarbonate, some other lubricants, right? Mucus can be slippery, but it's giving the saliva its viscosity. Notice this is a much thicker, much more viscous secretion. So notice when you look, it doesn't have just one duct. Notice it has multiple small ducts. Let's make these a little bigger. Multiple small ducts that come out and express right along the lateral aspect of the tongue. In fact, if we get this person to open up and say, ah, here along the lateral aspect, notice they only put 3 on this one, the last one had 4 on them. Along the lateral aspect of the tongue down there, we see multiple very short ducts to express that mucus out underneath the tongue. Remember this view, I didn't show it for this one because even here you don't really see it. But again, in the vestibule right here next to the second molar would be where that parotid gland would release its secretion. So even that you still don't see it, but we can see the relative where it would be. All right, and now we've looked at it histologically as well. And that leaves us with one gland. It's only intermediate in size, but the submandibular is the one that produces the majority of our saliva. Somewhere between 60 and 80% of your saliva is produced by the submandibular gland. It produces both serous fluid and mucin. That should tell you something right now before we even look at it under the microscope. If it is going to produce serous fluid, what type of secretory structures must it have?
Acini.
Acini or alveoli. Excellent. If it is going to produce mucus, what type of secretory structures must it have?
Tubular.
Tubes. Excellent. So notice what's going to happen here is when we look at this under the microscope, we're going to see a gland that actually has three things. It is going to have acini, it is going to have tubes, and it is going to have ducts. We're going to be able to recognize them. We're going to be able to know which is which. We're going to know the substance that we're going to make. We are going to know the functional classification, and without even seeing it under the microscope, the fact that it has both tubes and acini, what must its structural classification be?
Compound. Compound.
It's going to be a compound, but you got to give me all of it. Compound what? It's got both
Tubuloalveolar.
acini and tubes. Excellent. Tubulo acini or acinar or tubular alveolar. Either of those are fine. Tubulo. Which one did I write? Alveolar. Excellent. So notice we know all of this about it before we've even looked at it under the microscope. Awesome. Notice also it has one singular duct, and its singular duct is the one that releases right at the lateral aspect of the lingual frenulum. All right, again, if we look inside the oral cavity, right here and right here are those openings to the ducts of our submandibular gland, which produces the majority of the saliva. And some individuals are actually able to express saliva out of these ducts and propel it through space. And what do we call that? Come on, I know at least 3 of you must have been able to do it in high school. There you go, gleeking. I have no idea what the appropriate anatomical term for that is, but that ability to express the saliva out of that gleeking, right? That's the only term I've ever heard for it. That is the expression of that saliva out of that submandibular duct. Excellent. And not surprisingly at all, when we look at it under the microscope, lo and behold, we see 3 totally different things. Thing 1, thing 2, thing 3. What is thing 1? Duct. What is thing 2? Acini. What does thing 2 produce? Serous fluid. And what is thing 3? Tubule. What does thing 3 produce? Mucin. Beautiful. All right. So just to beat the dead horse because it's fun to do. What gland is this?
Sublingular. Submandibular.
Mandibular. Excellent. What is the substance it produces?
Mucin and
Serous fluid and mucin. In fact, in this case, we probably could get away with saying saliva, right? Because that is both parts of it, but I like serous fluid and mucin. Excellent. What is the functional classification?
Marrocrine.
Marrocrine. And what is the structural classification?
Compound. Tubulo-alveolar.
There you go. Compound tubulo-alveolar. Beautiful. Excellent. Just that simply, we have identified all 3 of our salivary glands histologically and answered any possible question we could think about for identifying for them. I guess technically, The only other question I could ask from one of these slides right here is, where does the duct express itself into the oral cavity? Because if by looking at the histology slide, you can figure out which gland it is, you should tell me where the duct expresses into the oral cavity. So this one would be just lateral to the lingual frenulum.
So all of them are expressed in the to the oral cavity, right?
Yes, but in different locations. Submandibular expresses, remember, just lateral to the lingual frenulum. The sublingual is along the lateral aspect of the tongue, and the parotid is into the vestibule, the superior vestibule, next to the second molar. Excellent. I promised you some histology today, and lo and behold, here we go. All right, questions on that? All right, I know it's a little bit early, but I think this is an excellent stopping point. We are not quite done with the oral cavity yet. We need to move on to the teeth next and talk about the anatomy and the function of the teeth. And then we can talk more about some of the physiology of the oral cavity from there. But I think this is a good stopping point for today. All right, any questions on any of that?
Can you repeat yourself where they express themselves, please?
Yeah, here, let's cheat and do this. Again, notice on the exam, actually, this isn't as good for that. Interesting. We know that this here is the, no, red. This here is the parotid gland. And the parotid gland has a parotid duct, which again expresses into the superior vestibule next to the second molar. All right. The submandibular gland has a duct that, notice, sneaks past the lingual, the sublingual gland, and expresses right out here, right next to the tongue. So again, we'll look at that other picture in the oral cavity, right next to the lingual frenulum. So we can recognize it and its duct. And then green, our sublingual gland has multiple ducts that we can see here express out along the lateral aspect of the tongue into the oral cavity. So here on this illustration, we can see all the glands, can't quite see all the ducts as nicely here. The lecture does show it a little bit nicer. So when we look at it here, notice again in green, they've shown us our ducts. So we can see the parotid duct, we can see the sublingual duct, which again comes up to here, and then all those little 5 little sublingual ducts that come here. And then also remember when we looked at it from this view, we can see that again, these over here lateral are the sublingual ducts, the openings of the sublingual ducts, and these are the openings of the submandibular ducts. All right. Questions on that? All right, excellent. We covered a lot of great material today. Excellent. A lot of anatomy, a lot of heavy lifting. And then we've got a lot of physiology coming up. So we're going to say, as we start working through all the organ systems, we still aren't done with the oral cavity. A lot going on in there. So we will finish that up and then continue our merry magical journey through the alimentary canal on Thursday. All righty. Excellent. Go spend wisely on this great national holiday that is Amazon Prime Day. Hopefully you have money left over at the end of the day. All right, take care. Have a good day. Be safe. Listen to the doctors, not the politicians. Vote. See you guys in 2 days.